that the world is aging rapidly. And in fact, in 2050, people with aged 60 years old will reach the 35% of the entire populations. So our question was, is it possible to rejuvenate senescent cells? Okay, so there are some strategies, and one of which is the use of cell secretome. So the cell secretome, what is? It is a heterogeneous pool of bioactive molecules that are secreted by the cells into the extracellular space, which are important for intercellular communications. In a cell secretome, we can find secreted proteins that includes, for example, growth factors, cytokine, chemokines, and non-protein components such as lipids, microRNAs, exosome, microvesicles. So our experimental design was to use young fibroblasts. So young fibroblasts were fibroblasts obtained from young patients with the age between 30 35 years old. We culture them and we collected cell secretome to use as a treatment in aged cells. Aged cells were fibroblasts obtained from centenarian patients. In parallel, we use the same treatment. So we collect cell secretome from young cells. We isolate EVs using chromatographic columns. We collected EV fractions, and we used EVs as a treatment in aged cells. We also treat aged cells with young cell secretome depleted from EVs, and as a control, we used aged cell secretome as a treatment. As a result, we detected a cell morphology change. So, as we can see here in this slide, young fibroblasts are characterized by a small dimension, so fibroblasts are elongated with a central nucleus, while aged cells are characterized by a bigger morphology. So as you can see, the treatment with the young cell secretome and EVs obtained from young cell secretome have an effect on cell morphology. So aged cells change their morphology, becoming smaller in size, while there was no effect in aged cells treated with young cell secretome depleted from EVs or with a control. We also analyze cell proliferation. So here we can see two different stainings. In blue, nuclei are stained for DAPI, and in red for PCNA, which is one of the proliferation marker. And once again, we detected the same situation. So when aged cells were treated with EVs or with young cell secretome, we have an increase in cell proliferation, and this cell proliferation level is statistically comparable to young cells, while this effect is transient, temporary. So as we can see from the graph on the right, we can see that at day 10 of culture, aged cells reduce their proliferation and come back to the original. We analyze also the effect on oxidative stress, in particular the accumulation of ROS and beta-galactosidase, and exactly the same situation. So EV's treatment and young cell secretome have an important effect in the reduction of these markers instead of the use of EV's depleted young cell secretome and aged cell secretome. Interestingly, the reduction in oxidative stress was also comparable to the an effect on gene expression, and in particular we focused on markers, senescence-related markers such as P53, P16, and P21, where we have exactly the same situations that before. So after the treatment with EVs or with the young cell secretome, we have a reduction in these markers until day 11, when the situation is exactly comparable to the untreated cells. And also for reactive oxygen species modulator ROM1 and mitochondrial activity markers such as TFAM, PDHA1, and COX4I1. While for the cell proliferation, exactly the same. So we have an increase in the cell proliferation rate after the treatment till day 5, where the proliferation rate starting to go down. At this moment, so at this stage, we observed this effect. So the hypothesis was that EVs have this effect. So what we did was to start in the characterization of EVs. So we collected EVs from both cell secretomes, so from young and aged cell secretome. We characterized EVs using TEM microscopy, and we saw that both samples contain EVs. We characterized also for Western blot for the typical markers, so CT9, CD63, and CD81. But the cytometric analysis confirmed that the presence of these markers was exactly the same in both samples. So, okay, both samples contain EVs. They were positive for the tetraspanin markers. So at this point, the question was, what is the difference between EVs obtained from young and obtained from aged cells? Okay, so we started to characterize also the content of EVs. So we were interested in two specific miRNAs, miR200B and C, and we observed that the expression level of these typical markers were different. So miRNA were higher in young cell secretome than in aged. Okay, so this was one of the difference, and we tried to custom out these miRNAs encapsulated in liposomes, and we transfected aged cells with these miRNAs. And what we saw is exactly the same effect that I described before. So we have an increase in cell proliferation and a decrease in senescence-related markers, mitochondrial activity, and reactive oxygen species. So probably this effect in cell rejuvenation is due to the presence of these two miRNAs. Okay, so in conclusion, what we can say is that this experiment demonstrates the possibility to counteract age-related biomarkers in senescent cells, and this effect is demonstrated with the use of EVs derived from young cell secretome. And if we depleted young cell secretome from EVs, we did not detect any effect. And also, probably miR200B and C together may be responsible for this rejuvenating effect. Altogether, these results suggest the implication of signaling pathways that may counteract age-related molecules via soluble factors containing young systemic environment. I would like to thank all the supporting fundings that were very, very important for this research, my supervisor, and all the lab team. And of course, I would like to thank you for your kind attention.
Thank you, Sharon, for the informative talk. And now the floor is open for the questions. Is there any question for Sharon?
Thank you for sharing your data. How did you isolate EVs?
We used chromatographic columns. So we collected young cell secretome with centrifuge, and then we put the supernatants into chromatographic columns.
Okay, so size exclusion.
Yeah.
I see. Okay, thank you.
You're right.
Because I was wondering if there could be other components other than vesicles, so that's good. Thank you.
Thank you.
Sharon, thank you very much. You also showed that there is a difference in the number of the extracellular vesicles. I think
Yeah.
one of the graphs was showing that. It's not very clear for me how many fold difference there are, because, or is it 10 times more?
Yeah.
In the aged one?
I'm sorry.
It is 10 times more in the aged one. And then that means that when you are somehow incubating the cells with the aged ones, they are, you have also a higher amount of the EVs.
Yeah, we detected a difference in EV's concentration in the samples. So aged conditional medium was richer in EVs, but the difference was in the content. So the Mirna content was different, and probably this was the reason why we have an effect in when we used EVs derived from young cell secretome instead of aged. So yeah, the content is completely different. So aged cells release more EVs, but the difference is in what there is inside EVs.
Have you looked also at the profile of the EV size, that if there is any changes in the size of the EVs that you are using?
We try to measure the size of EVs, and we did not detect any different difference. You're welcome.
Thank you very much. And we have a token of appreciation for you. So Gayandi will give the next speaker's introduction.
Thank you. Hello. Our next speaker will be Professor Tiziana Brevini, and she's a full professor of anatomy and embryology at the University of Milan, Italy. And her main area of research focuses on the understanding of cell differentiation and commitment, epigenetics, paracrine and mechanosensing related controls in mammalian cell fate and embryo. Today, she will be talking about synergistic interactions among molecular effectors and biomechanical cues to drive cell fate and differentiation. And Professor Brevini, the floor is yours.
Share with you results obtained in our lab in the last few years. We have a distinct interest in understanding the mechanisms that are driving cell fate and differentiation. Most of our work is done in vitro, so I think I should point out this. And we have a distinct interest in understanding and characterize the cues deriving from mechanosensing related responses in cells, of course, combined with molecular effectors. So you won't be listening too much about exosomes from me, at least today, but rather we will be discussing about the extracellular environment and a little bit of point of view I have in terms of predictivity of in vitro models that we use, eventually even to characterize EVs. And I would like to start reminding you about the pioneering work carried out by Mina Bissell. Mina Bissell, she was working at Berkeley University in California, and she firstly demonstrated the key role played by the extracellular matrix in mammary gland cultures in vitro. And she demonstrated that she was able to maintain the correct phenotype and functionality, whether she was co-culturing mammary gland cells using extracellular matrix. So this is officially considered the start of the organoid era, because she firstly demonstrated the importance and the contribution of the extracellular matrix and the relation of cells with the environment in order to maintain a correct function in cells. Of course, since then, quite a large numbers of models and 3D options have been produced, and we know we have organoids and 3D cultures of most every tissues, which have been produced by several labs in the world. And I would like to just discuss this a little bit more with you today, if you allow. So we know that when we culture cells in a monolayer, which is the most classical way to approach the situation, cells do acquire a morphology which is completely different from the one that they would have in a normal tissue with a normal architecture. They tend to flatten, and their contact, the cell-to-cell contact, is significantly reduced if we consider a normal situation. So this obviously does affect cell behavior. And as an example, for instance, the problem is that we can end up with models that significantly deviate from the normal and the physiological condition in vivo. And for instance, this change in behavior can be very misleading in interpretation of results and data. And just an example is given by the astronomical cost which is required to produce new drug. Because, of course, all this difference becomes evident just in the preclinical phase, since we trusted our model until that. So this obviously has to draw our attention to the fact that we need reliable models that have a higher predictivity. So what strategies can we adopt in this sense? And so I will just try to give you a very brief overview, and we'll just tell you about our experience about this. So, of course, a nice way to approach the problem is to use 3D culture systems. So the use of scaffolds, for instance, will, as in this shots, you can see that if we use a polystyrene thick scaffold, we allow cells to penetrate. And mostly important, we allow cells to maintain their three-dimensional aggregation and organization. And this will completely change the situation from what we saw in our previous slides, where we saw cells flattening and acquiring a completely different way to engage in cell-to-cell interactions. Of course, a very important thing is the stiffness of the scaffold we select. Well, the accumulating data right now are clearly pointing to the fact that each tissue in the body has its own stiffness. So when we deal with a neural system, we are dealing with a certain stiffness. And when we are, for instance, dealing with bone and osteoblasts and osteoclast, we will need an environment which is completely different, much harder. And as you can see here, there is an increased stiffness in tissue. This stiffness is measured, expressed as Young modulus of a tissue. So each tissue in our body has a specific Young modulus. And we have to keep this in mind, because when we grow, for instance, neuron, and we try to differentiate, I don't know, neurons from stem cells, basically, if we use a plastic device, mind you, this is the position where Petri dishes and flask and stuff like that do position in the stiffness scale. We are basically telling our cells that we want them to differentiate towards neural phenotype using our growth factors or whatever soluble factors that we chuck in the culture medium. But at the same time, we are telling neurons that in terms of stiffness, we would like them to turn into bones, for instance. So this is, of course, very confusing, and it's also a very, we are basically making our life very difficult and our cells' life very difficult as well, because we are giving our cells extremely contradictory information. Well, so this is another point. Selecting the right scaffold and selecting the right stiffness in the scaffold we are using. But how does the cell know what's going on? So why is the cell sensing this? Of course, there is a lot of work going on in this area, and there are a number of signaling pathways that have been characterized in the last few years. I will just give very briefly I will just say a couple of words about probably the most classical and most known one, which is the hyposignaling pathway. The hyposignaling pathway is a very complex pathway, but to cut a long story short, basically there are two actors involved in this, and are the factors YAP and TAZ, and usually YAP is used as a marker of this. And what happens is basically that cells is informed about the stiffness and the environment through YAP, and YAP will phosphorylate and be compartmentalized to the cytoplasm in response to a certain stiffness, and the compartmentalization of YAP to the cytoplasm will instruct cells to differentiate and not to proliferate, basically. In contrast, in response to a different stiffness, YAP will not phosphorylate, will translocate to the nucleus and will interact with further factors such as MAP24 and TED, and a number of different factors. And the result of this will be proliferation. So basically, for instance, when YAP is confined to the nucleus, cells will self-renew and will avoid to differentiate. So this implies that we can basically influence cell behavior and cell fate decision, driving stiffness and driving cells to choose to translocate their own nucleus either to their own hyposignaling related factors, either to the nucleus or to the cytoplasm. So you can, here in red, you can see nuclear exclusion and cytoplasmic compartmentalization or the other things, simply by that. And there are a number of papers that show this interesting kind of data. Okay, so basically selecting specific culture approach, we can try to drive cells towards a certain decision. And I will just give you a couple of examples here. For instance, in these experiments, we use microbioreactors, and these microbioreactors were prepared using polymer, which is hydrophobic, and it's called polytetrafluoroethylene. I will just call it PTFE from now on because it's quicker. Anyway, this PTFE allow us to create microbioreactors where we encapsulate cells, and it's gas permeable, so cells can make all the necessary exchange, but it will provide cells with certain information in terms of cell aggregation and organization within the system. And so we can just prepare this microbioreactor and float them on the medium. And basically cells will be living in a kind of microplanet, doing whatever they want to do, and simply exchanging information with the medium. In this case, we were trying to prepare inner cell mass-like structures, so the inner cell mass within the embryo, and we were interested in maintaining pluripotency in this microbioreactor. By the way, these experiments were carried out in collaboration with my colleague, Giorgia Pennarosa, who's not here today. And as you can see in this slide, we were using here OCT4 GFP positive fibroblasts. So basically, these were fibroblasts that were reprogrammed to pluripotency. And when they become pluripotent, they also turn green because they start to express OCT4. So this is a nice user-friendly way to monitor cells, and you know that they are pluripotent if they turn green, basically. So as you can see, you can see this nice green emitting ICM-like structures that were prepared using this encapsulation system. And of course, I'm not going into all the details, but all these cells express the pluripotency-related factors that are distinctive of ICM and pluripotent cells in general. So we go back to the hyposignaling story now. And as you may remember, in order to keep these cells in a pluripotent state, we want to have YAP translocating to the nucleus, okay? So because when YAP translocates to the nucleus, it will bind SMAD2. SMAD2 will also translocate to the nucleus, and it will activate OCT4, NANOX, OX2, all the factors that are distinctive of pluripotency. And the other way around, if we keep YAP outside the cells. And this is exactly the situation. When we reprogram cells, as in this case, we can see that if we culture cells in plastic, we get the results. I'm not saying that you don't get the results. The cells are reprogrammed and we get all the expression and so on. But this is very quickly lost. And by day 7, all the YAP goes back to the cytoplasm, which basically means goes back to non-pluripotent condition. In contrast, when we use the microbioreactor aggregation system, we can keep a nuclear compartmentalization of YAP, and therefore we can keep pluripotency for a much more extended period, suggesting that using the appropriate stiffness or the appropriate rearrangement, we can maintain cells in a certain phenotype and in a certain differentiated state. I'm not going too much into this, but basically the story here is that to show this, we knocked down one of the two elements. We decided to knock down TAS. And if we knock down TAS, we have no nuclear translocation. Of course, we don't have TAS because TAS is not there anymore. But we also have no nuclear translocation on SMAD. And therefore, as you can see here, purple bars, pluripotency-related markers are down. So basically, we very quickly lose pluripotency. So suggesting again that the ability to keep cells in a pluripotent state for a long period in the presence of a microbioreactor is due to the presence of the microbioreactor. Since when we kill the role of the microbioreactor, we lose the pluripotency as well. Of course, these were very exciting experiments to us. We are mainly an embryology lab, so being able to create ICM-like structures in vitro was very appealing to us. And in the meantime, I have to say we were also working in a parallel work to reprogram cells towards a trophoblast-like phenotype. And so together with, again, Giorgia Pennarosa and Sharon, which you just heard was our previous speaker, we decided to put the two mainly data together and to use the system to create epiblastoids, which are organoids that mimic in vitro the behavior of blastocysts. So that would be a very advantageous way to study certain mechanisms taking place in the early embryo, of course, bypassing the ethical problems that we would encounter by using real embryos. And so this is the scheme of the experiment. We reprogrammed the same cells. So mind you, we were starting from the same cell population, which were fibroblasts. And then we reprogrammed fibroblasts towards ICM-like structures with the system I just told you about. And then in parallel, Sharon was differentiating cells towards trophoblast-like cells with a well-established protocol. And then we would put the two populations together and once again encapsulating them in an appropriate system, culture them in agribel, which are dedicated, embryo-dedicated vessels, and with a stable, providing a stable concentration of a number of different factors, we would be able to create what we called epiblastoids. And this is what we get. And so you can see these structures look very similar to embryos with the cells exactly in the right position, the trophoblast and the inner cell mass and the two populations together. And I think this is one of the possible strategies that have been used in the last year or so to create embryo-like structures for in vitro studies. And I would like to draw your attention to the fact that when we are doing these reprogramming experiments, we are basically erasing the epigenetic, temporarily erasing the epigenetic memories of cells and reprogramming cells towards a different phenotype. This takes place through several different mechanisms. And in particular, in this case, we were decreasing the DNMT, de novo methyltransferase activity, in order to basically reduce methylation in cells. And at the same time, we were activating a family of enzymes which are called the 1011 translocation enzymes, or TET enzymes. And these enzymes are in great interest recently because they are mainly the factors that are activated by the Yamanaka reprogramming protocol. So if you want to reprogram a cell, and it has been shown that you have to have an adequate amount of expression of the TET11 family. And it has also been shown that if you knock down 10, or if you have a TET-null mouse fibroblast, you cannot reprogram a fibroblast like that. It's unreprogrammable. So they also showed that if you, however, treat cells with TET downstream miRNAs, you get the possibility to reprogram cells. So you can bypass the fact that the fibroblast is not expressing endogenous TETs. And interestingly enough, the downstream miRNA, TET downstream miRNAs are miR200B and C, which are the same miRNAs that we were actually seeing in rejuvenation, and Sharon told you about. So this was very interesting to us. And of course, we are not the first one connecting aging and rejuvenation and self-renewal. There are a number of papers by Belmonte and by a number of groups that show a strict correlation between the possibility to drive cells towards a young stage, an even younger stage, and even a non-committed stage and pluripotent stage, and the other way around. So they basically identified a very common pathway there. So what came to our mind was, once again, with Sharon and Georgia, to reprogram cells and repeat exactly the same experiment I just told you about in the production of epiblastoid. But in this condition, we decided to use directly MIR200 and B and C instead of a normal reprogramming protocol. And I'm not going into details, but we exactly got the same results, and we got exactly the same kind of organoid. So I think this is a very stimulating kind of information, and I think it's probably worthwhile to further investigate this, and we definitely will. So just to close this part of my talk, I hope I convinced you that biomechanical cues are playing an important role in driving cell fate. Of course, they are not working alone. There are a number of numerous factors involved in this, but we have to keep that in mind when we plan experiments. And probably a lot of process that we are trying to understand would be easier to monitor and follow by selecting the correct environment for the cells. Of course, we have just seen that epigenetic cues are there and soluble factors, and so the NMTs and TETs and different families of MIRNA are there, and we should keep this in mind when we are playing around with cell fate or cell phenotype. I would like now to go back a little bit to what I began my presentation with, so the role of the ECM. And of course, the ECM is basically what cells sense in the real life. So it's their own 3D environment. So I think that the ECM knows. We have to look at the ECM and try to mimic what they sense in that respect. And indeed, this is what we are trying to do, and a lot of laboratories are trying to do, using hydrogels and using all sorts of polymers in order to offer cells to a certain information. At the same time, another approach can be to add components of the ECM, such as adding collagens and other stuff that is nowadays used for in vitro models. So the ECM, the emerging role of the ECM, there are, of course, different ways we can mimic ECM, as I said. The idea that we are mostly adopting is to use directly decellularized tissue. So basically, we can use tissue and we get rid of all the cells in the tissue. And so we are left with the pure ECM. And so by doing that, we make sure that we don't need to recreate an environment because the environment is already there, ready for us. The ECM is very interesting because it provides the correct mechanical integrity. And interestingly enough, it's highly conserved throughout species. So there is basically no immunogenic response. So this means that we can use a decellularized heart and we can repopulate it with human cells. And there won't be an immunogenic response. So it is not surprising, having said this, that there are a big interest in many laboratories to try and create a situation where we, for instance, take a liver lobe here, you can see, and then we can perfuse this tissue in an appropriate way, and at the end of a couple of days, I wouldn't use at the end of the day, sorry, we end up with a structure like this, completely transparent, completely devoid of cells, and you can see all the remnant of the vessel beds and everything. So it is obvious that, well, there is a basically a whole body of information about the way you can proceed to decellularize structures, and it's just your choice. And there is a very distinct variation from tissue to tissue, so you can just basically play around. And usually the communal aspect will be to use a strictly physical approach in the first step, which can be, repetitive freezing and thawing, or a different approach, high hydrostatic pressure. This is different labs. And then an enzymatic step using different detergent, and then a chemically more harsh step. So then once we have the decellularized structure, we can either use the decellularized structure the way it is for further experiments, or we can solubilize the structure, make what it is called bioink, and then use this bioink to 3D print a structure of our interest. So it's really our choice. Of course, we will need the appropriate 3D printer that keeps and doesn't, destroy or denature the nature of the ECM. Okay, we basically did a lot of work on the ovary, because our target was to end up creating an artificial ovary, and we have a distinct interest for especially young oncological patients that might have troubles, and that could be the very perfect niche to keep their follicles intact during chemotherapy and waiting to see whether they will then decide to have kids. Okay, so when we started up this work a few years ago, only decellularization of fragments was available. Nobody has had decellularized whole ovary. And here, this is the work by Matteo Ghiringhelli, who was a PhD in my lab and is now engaged in a postdoc in Israel, together with Georgia. And they managed to completely decellularize ovaries of different species. And of course, in our project, the idea was to create an artificial ovary. So the next step was, once we knew that we didn't have toxicity, apoptotic process was very low, and the morphology was kept. We were pleased with that, and so we decided to repopulate this scaffold. And we were repopulating with ovarian cells. So this was step number one. And so, because you never know whether cells will like your scaffold. So you have to check whether once you seed your cells will stay there or will prefer to go somewhere else or die simply. So we could see that there was both an increase in cell population and cell density. Not only that, but when we then monitor the main markers for ovarian cells, which was present before seeding in cells, we could see that the scaffold would offer the environment that really cells did like, which is not surprising because they are home in a way. And even after 7 days, cell would maintain in a statistically comparable way the expression of the genes that we were looking at. So this was interesting. This was a first step. And then in our idea, the next step was to use high plasticity cells, so reprogrammed cells instead of ovarian cells, because this would give us the information about the possibility to inform cells and drive cells towards the ovarian differentiation once we were using undifferentiated cells. So the ability of the ECM to drive cells towards the correct differentiation. So we here used reprogrammed cells, and we were basically repeating exactly the same experiments, looking at what was going on. And so you can see here, so their high plasticity cells are here depicted in green bars, and the yellow bars are the ones from the ovarian. So just to compare what we saw in the previous slides. And you can see that at the beginning of the engrafting, cells would express the pluripotency markers as expected because they were pluripotent. And then they quickly lose pluripotency related marker expression, but they start to pick up the factors that are distinctive of the ovarian tissue. So this was very exciting, of course, because it's somehow telling us that we are going in the right direction, and that ECM is interacting with cells and is telling cells, look, you are in an ovarian environment and we would like you to behave the way we want. So this, of course, you know, we are working a lot on it, and I think it's a very promising area. And it can be applied, of course, for transplantation, for instance, because you can imagine you can use patient-specific cells and recreate patient-specific organ in the long run using a very specific scaffold, maybe taken from, you know, a porcine or whatever, depending on the size you need. So, of course, this is a kind of science fiction, but I think it's already been done, and there are artificial organs out there. So, yeah, right now we have a project which is strictly related to the one that Sharon told you about. Is about aging, and Teresina De Iorio, who's also a PhD in my lab, she is working at ovarian bioscaffold, and she's comparing the differences in scaffold obtained from aged ovaries and young ovaries, and this is related to, you know, all the information we need about changes in lifestyle, especially in women, and, you know, everything, all the information that are related to reproductive aging. So this was very appealing to us, and she could show that the scaffold would maintain the changes that we detect in the native tissue. So in aged scaffold, we would have a disproportion, the same kind of changes in terms of collagen and elastin that we see in the native organ before decellularization and the other way around. So this is, again, a very promising scaffold where we can then use young cells and aged cells and play around with exosome and Myrna and stuff like that. So this is another model we are working at. So just to close, I will just go back to what Sharon was telling you, and I will, of course, skip all the part where she told you about the role of AVs and the rejuvenating effect of AVs, because she very nicely told you about it, and the possibility to isolate, or to identify a contribution, of course, together with many other factors of the MIR200 family. What I could add that what you already saw about the effect of the MIR200 family in terms of rejuvenating, as you could see, what Sharon told you is that the rejuvenating effect is very evident, is statistically interesting, but it's quickly lost. And within 1 week, cells go back to what they were before the exposure. Well, here again, similarly to what I showed you with the ICM, when we use a young ovarian scaffold, we can extend and stabilize the effect. So once again, we can see that using the correct scaffold and the correct stiffness and the correct environment, we are somehow instructing cells to keep that phenotype, the acquired phenotype. And indeed, by using you can see here in red bars, the rejuvenating effect, which is quickly lost, blue bars, in the absence of the scaffold, is here maintained, red bars, when we engraft the rejuvenated cells in the scaffolds. Okay, so I hope I just gave you a very quick overview of the different 3D approaches that can be used. Of course, there are many other there. I decided to focus on these mostly because these are the one I like better, and these are the one I know better. And also, of course, the choice of the scaffold and of the 3D culture condition we select is basically influenced by the scope of the experiment, what we want to get out of the experiments. But I think that the very important thing is that the options that are now available are really offering us something which was even, science fiction until 10 years ago in terms of what we can create in vitro using a 3D approach, trying to replicate physiological environment as much as we can with a very high efficacy and efficiency. And this, of course, has the great advantage at the end of the day to reduce the use of experimental animals, because it will give us information with reliable data, reducing the number of animals that we need to confirm the translation from the lab to the real life. And with that, I would like to acknowledge the contribution by the very many people that were involved in these experiments, and to thank, of course, the funding bodies, and last but not least, to thank you for your attention.
Thank you, Tiziana, for the very informative talk. As we are running out of time, the floor is open for just 2 questions.
Thank you, Tiziana. It was very interesting talk. I'm a bit interested on the epiblast work that you presented. So may I know what was the percentage efficiency-wise when you reprogram the fibroblasts into the stem cells, and then you again convert them into trophoblasts? What was like the efficiency that you got there?
Okay. Efficiency in reprogramming depends greatly. We have high efficiency because chemical reprogramming is a completely different story from the Yamanaka kind of reprogramming. And indeed is a kind of progression of the idea. Yeah, I'm not saying we are better than Yamanaka. I'm not saying that. But what I'm trying to say is that, of course, 20 years or so had to understand better what's the process involved. It greatly depends. We can get around 60% to 80% of cells that enter a plasticity state, that we will have a cut again, but this doesn't depend on the reprogramming, but on the differentiation during the differentiation state. So depending on the kind of tissue, you can get between the 30% and the 40% of the final cells you want. I don't know, pancreas or whatever. You can play around a lot with efficiency if you play around with cell cycle, because your reprogramming factors can be DNMT, HDAC, or whatever you are using in terms of epigenetic changes, do affect cells in very specific cell cycle phase. So, for instance, if you want to decrease methylation, you have to catch the cell during the S phase. Okay, so if you synchronize cells before exposing cells to the reprogramming factors, then you might end up with the 95% of erased cells. That doesn't mean that you will have 95% of final phenotype cells. Of course, if you have a very heterogeneous population, 30% in S phase, 40% in G1, and so on, or G2, then you only catch the cell which will respond to that specific mechanisms that might be G2 or S or whatever. So it's very, you have to think in advance what you are, to put the two aspects together.
And one more thing in connection with that. So have you ever had any data on single cell resolution level after reprogramming? Like, because I know in my previous lab they were doing the same but using the stem cells. But when end of the day, of course they were able to get the trophoblast cells, but end of the day when they did the single cell sequencing, then they found, well, like less than 3%, 4% are actually representing the real transcriptome and all that.
Sorry, I'm not sure if I understood exactly what you're asking.
No, I mean, once you reprogram, I mean, once you got your trophoblast cells after, reprogramming the fibroblasts and everything, once you have, before you do this epiblast formation, have you ever had any single cell resolution level, how many or what percentage of cells really, representing the trophoblast? Because that's a cliche like thing everyone is, fighting right now. Yeah.
Well, I think this is another very open aspect because, of course, once you are reprogramming cells, you then want to have a certain efficiency and a very distinct phenotype.
Yes.
And you have to deal with different factors. One is using scaffold or whatever. You can simply use a correct stiffness. If you want to do pancreas, you have to make sure that you are having an ECM around or whatever, a scaffold that has the Young modulus, which is distinctive of pancreas, which is 4 kilopascal, by the way. But anyway, so you will select the correct matrix. And the other thing you have to make sure is that you have a steady state concentration of the differentiating factors. For instance, we use a lot activin A for certain experiments. And we found that if we add activin A will be in the medium for a certain number of hours. And then the amount of activin A is quickly decreasing. So those cells that are differentiating in that time window will not have the appropriate amount of activin A. So what we do, for instance, we encapsulate factors in pods, like the one you put in the washing machine, okay? So you have pods with activin, pods with all your growth factors, and the pods will ensure a slow and continuous and steady release. So, there are hundreds of things that you can do, and all these factors will affect greatly the efficiency and the final results. And this is why you have big variations from laboratory to laboratory. It's not only cells, it's not being sloppy or super good. It's basically dissecting all different aspects that you have in cultures.
Thank you.
Thanks for the great talk. So have you thought about the connection between mechanobiology and EV secretion? Right, so we know that cortical forces around the cortex, right? So the plasma membrane can influence EV biogenesis. And to some extent, I'm not quite sure if this actually can also affect cargo loading, right? Into this extracellular vesicle. And I understand that this was not an EV-centric talk, but considering your interest in mechanobiology, I was wondering if you've ever pondered this connection and how that might impact.
Not aware, but I think this could be a completely open area. I don't know, Alireza, if you would agree with me, or probably you, because, well, I don't want to go into, but for instance, another very, very, very key aspect is what they call macro crowding. And this was a very interesting question you posed before. Because according to the number of molecules, of course, you have an osmolarity, you have a number of factors, and the macro crowding that you have in a medium is greatly affecting the response of the cells. So, for instance, let's consider H cells. We know that H cells release at least twice or 3 times the amount of cells exosomes that are released by young cells. And this is a very characterized pattern, which is related to aging. Okay, so of course these exosomes are containing inflammatory, molecules and all sorts of molecules that are clearly related to senescence rather than to being nice and young and so on. So, and apparently, why do they have so many? So I cannot possibly think that it's just a matter of composition. It must be also something related to the crowding that cells experiments, okay? But I don't think there's anything which has been studied. I'm not aware of it. Maybe laboratories are working at it. So what I'm trying to say is that probably there are factors that, of course, are related to exosomes that are greatly changing the way cells sense the environment. And probably the combination of these factors plus the stiffness, plus the 3D rearrangement, plus the way cells are organized in a kind of condominium and so on. All this aspect will obviously affect cell responses, but we are not used to consider this. We usually focus on either transcription factors or EVs, and we completely forget that we are, messing around with a number of other aspects that are probably there.
So my pure guess is that the rheological environment of the tissue would certainly affect EVO biogenesis, right? So I'm not really looking at the contact of the basal membrane and the ECM, but if you look on the apical or even at the junctions, right? So for you to be able to successfully produce extracellular vesicles, there are a certain number of sort of criteria that you need to meet before you can actually release, say, a microvesicle, right? So I think there again, tensile forces or compressive forces on the plasma membrane will bias those output.
Yeah, I think that we need to combine so many, information, but, we will make it slowly and slowly. Probably, maybe we won't make it. Somebody else in the future will make it. But for sure, the more complex we manage to be, the closer we get to the complexity of the system. Because I'm sure that, we have a very schematic and simplicistic way to approach something which is rather more and more sophisticated and complex.
With the interest of time, thank you very much, Professor Tiziana.
Thank you.
And we have a small token of appreciation for you. So before our coffee break, the last speaker for the morning session, it's Professor Suresh Mathivanan. He's from Department of Biochemistry, La Trobe Institute of Molecular Science, La Trobe University, Australia. He'll be joining through online today. Let me take a few minutes to give a small introduction to Professor Suresh Mathivanan. So he undertook a PhD in proteomics and bioinformatics at the Institute of Bioinformatics, India, and then Johns Hopkins University, USA. So after his PhD, he joined the Lund Institute for Cancer Research, Melbourne, Australia, and later moved to La Trobe University. Currently, his laboratory is focused on extracellular vesicles, their role in cancer, and intercellular communication. Adding to that, he has authored over 118 articles that are cited more than 35,600 times. He has been listed as a highly cited researcher by Web of Science for the last 5 years in 2018 to 2022. So his lab has contributed key studies in biochemical characterization of EVs as well as the bioinformatic capabilities. So for us, his today's talk is about extracellular vesicles in cancer progression and therapy. So over to you, Professor Suresh Mathivanan.
Thanks for the introduction. Can you guys hear me and see the slides?
Yes, it's okay. Thank you.
Okay, thank you. Thanks for that. So thanks for the introduction, as well as the invite to speak. So Today, I'm coming pretty much from Melbourne, Australia, and then again in La Trobe University. And today's talk is going to be focusing on extracellular vesicles in cancer progression and therapy, right? So before going on to the topic, so as a lab, we are a lot interested in extracellular vesicles for the last 13 to 15 years. And when we talk in the context of extracellular vesicles, we are interested in various topics like chemoresistance, cancer therapy, where we use extracellular vesicles as drug delivery vehicles, as well as we try to study the role of EVs in metastasis and cachexia, as well as to understand their role in biogenesis, and also have some bioinformatics capability in terms of development and maintenance of Exocata, Vesiclepedia, and Fundridge, right? But we also have some non-EV projects as well. But however, today's project, I'm going to present on milk EVs and their role in cancer progression. It started like a side project out of curiosity, and then later on it developed into a bigger theme in our lab, and that's what I'm going to start talking about it today, right? So for the purpose of this talk, I'm just going to call all these EV subtypes into extracellular vesicles. I'm not going to look at what are the subtype that I'm working with. Rather, it's a collective population of EVs, small EVs, large EVs, everything included together. Again, for the purpose of this talk, again, for this conference, I'll skip this slide, but what it represents is the effect, the video from NIH consortium saying that EVs have the capability of changing the phenotype of the recipient cells, depending on the context, that in a quick time or in a time-dependent way, depending on what context we are studying. But however, in the context of cancer, there's a lot of landmark studies around there. But what we have done as a lab is we've studied the role of EVs in the transfer of chemoresistance, where we looked at some sensitive neuroblastoma cells and then the resistant neuroblastoma cells, sorry. So we collected the EVs from them, and then we were able to show that these can transfer the aggressive phenotype to the sensitive neuroblastoma cells, thereby rendering them resistant to chemotherapeutic drugs. Similarly, we have also shown that EVs can transfer oncogenic mutant proteins, in this case, mutant beta-catenin, from one cell to the other, especially in the tumor microenvironment, where you have a population of cells with varying mutations, and these EVs can kind of keep the proliferative signal maintained in a tumor microenvironment and alter the tumor microenvironment in favor of cancer progression, right? So this talk, right? So this particular study started in 2012, almost a decade ago, where this particular paper was published in 2012 by a group in Nanjing University in China, which was published in Cell Research, right? So what they proposed was, when an individual is consuming rice, right, they were able to pick rice RNA circulating in the serum of the people who consume that rice, right? And then they went on to propose that this rice RNA was not only bioavailable, it was able to change the gene expression in several tissues, and thereby was potentially able to change the phenotype of the host organism, right? And they articulated this MIR168A as one of the important one. As you can see, this exogenous plant MIR168 can target gene expression in a way. So that story created a lot of interest at the time, and then it also created a lot of debate. But however, the authors hid behind the fact that the microRNA is able to survive the gut through these EVs, and they're resident in the EVs. And once they are in the EVs, they can either enter the systemic circulation via transendocytosis or via paracellular translocation, right? So I'm just going to go to the pointer option. They can either go via paracellular translocation or through transendocytosis. So this was also proposed by us in a review in milk about milk EVs, but however, there is no evidence, no experimental evidence to show that the EVs can go via transendocytosis or via paracellular translocation in vivo, right? So at the moment, it's all speculations in a way. But however, the hypothesis that the RNA or the EVs from a diet can survive the gut was refuted by other people. Like, for example, this is a paper in Scientist asking that plant RNA paper was questioned, right? Because you can imagine there's a lot of gene regulatory boards where they're looking at genetically modified organisms, and these guys were petrified when they're allowing these products to be consumed. And then they were like, okay, now we modify the organism. Are they able to survive the gut, and what repercussions would these have, right? So in a way, there's a lot of studies which followed up. So you can see several studies which refuted the hypothesis. Again, you need to be much more careful to refute a hypothesis because you need to have a lot of controls, especially this particular article in Nature Biotech, where they deleted the specific microRNA in the mice, and then they fed that mice with that microRNA enriched diet, and they were unable to pick that in circulation or in serum or in plasma. So thereby they were saying that we could refute this hypothesis, thereby this is not going to happen, right? However, at that time, I had a discussion with my collaborator who was interested in bovine milk, and we thought, okay, let's look at bovine milk because that is one beverage which is consumed a lot by the world. And when you look at the amount of EVs that is present in milk is huge. And it's estimated that an average human consumes 170 ml of milk a day, right? So we thought, okay, let's ask some simple questions to see whether this hypothesis or this concept is true. If it is true, we'll follow it up. If it is not true, we'll probably get out of this project and look for something else, right? So the question that we asked is, do EVs in bovine milk have a functional role? And first of all, we wanted to ask, can they survive the gut, right? And further to that, we asked certain questions in terms of, can these EVs survive the gut and retain the system and thereby be distributed to other organs? Do these EVs play any role in mammalian physiology or well-being? Do they have any immunomodulatory roles? As well as, do they have any role in cancer progression, provided if they can survive the gut, right? So as a cancer lab, we thought, okay, we can look at the role in cancer progression as well. So to isolate EVs from bovine milk, we adopted the methodology that we previously published in 2019, 2020, where we used a series of differential centrifugation coupled with ultracentrifugation, and then separate this EV by optiprep density gradient centrifugation, and then used for following analysis, right? So this is the data here. So now what we did is we collected EVs from both raw milk and commercial milk. Raw milk here is what we got from the farmers. No pasteurization, homogenization of any kind of processing was subjected on these milk. And these are pooled milk from several cows, and then we got several batches of them. Same way, commercial milk was what we got from the local grocery stores. And we got 5 to 6 brands of those commercial milk and pulled them together, and then isolated EVs from them by optiprep density gradient centrifugation. You can see that the EVs which we isolated were positive for this EV enriched protein called TSG101. But as you can see, when you look at the whole milk, casein was abundant, but when you look at the commercial milk EVs, casein is depleted. However, TSG101, the luminal enriched protein, is highly present in this EVs. You can also see by transmission electron microscopy and NTA that there is a mixed population of EVs in the preparation, both from raw milk and commercial milk, as well as there are EVs up to 300 nanometer in size here, but the peak size is somewhere between 145 and 135 in a way. So next, we asked a question in terms of what is the cargo of these EVs? Does it change again, based on the processing parameters. Again, there's a lot of variables here, but to cut the long story short, what we found out was there is indeed difference in the protein and the RNA cargo of the raw milk and commercial milk EVs. But however, there is also some components in, despite the different batch of milk, different cows, different food that have been fed up, and as well as all the processing that has been undertaken, there is still a lot of proteins and RNA which were common between them. But however, there is also a lot of proteins and RNA which are different between the commercial milk and the raw milk EVs. So then we asked the question, can these EVs survive the gut? So what we did is we took the EVs, the milk EVs, labeled them with this lipophilic dye called DIR, and then orally gavaged them into the mice. So we wanted to mimic if someone is drinking milk, and we just gave physiologically relevant dosages to this mice, and then wanted to check if these EVs can survive the gut and reach other organs, and then we wanted to visualize them. And here is the data. So after 24 hours, you can see PBS alone, there is no signal. But if you look at the raw milk EVs, after 24 hours, you can see there is signal in heart, liver, lungs, kidney, spleen, and the GI tract, right? But when you look at the dye alone control, you can see there is dye alone control is pretty, like say the faint, because the dye alone can diffuse into the intestine and maybe other organs as well. But when you sonicate the EVs and disrupt the membrane, you can see that is similar to the dye alone control, and then the high signal is lost. And you can see this is the quantification of those data. What it suggests is EVs, the membrane, the lipid bilayer of the EVs is important for them to survive the gut and reach multiple organs in the mice, right? Once you sonicate, it's not able to pass the gut, as well as the dye alone can diffuse the gut. So you have to be very careful with such of this data. As I told you, the dye alone can diffuse. I was not 100% confident with this data. I was a bit skeptical. Said, okay, let's look at this in different perspective. Can we ask this question in a different way? And if we can answer this, we may be a bit more confident with this, to see whether we can pick any of these proteins, the bovine proteins, in other organs. So what we did is we did try a lot of Western blotting and other techniques, but we were unable to get any bovine specific antibodies. They're always cross-reacting with mice as well, the mouse as well. So what we ended up doing is we went for proteomic analysis, where we took the treated liver and the untreated liver. We subjected them to label-free proteomics, and then we looked for bovine-specific proteotypic peptides, right? So what I mean by proteotypic peptides are, these are peptides which are uniquely identified by the bovine genome, and it doesn't hit any other genome, not bacteria, not mice, not anything else, right? So we were able to identify more than 125 proteins like that, and then we look at the coverage, it's almost like more than 80% as well, suggesting that they're not fragments which are going there. So then I was a bit confident with this data, suggesting that yes, these cargo are indeed reaching to the other tissues. So then we said, okay, maybe if you look at the functional role of these EVs, we'll be even more confident. So what we said is, okay, you look at the role of these milk EVs in cancer. And then for that, we use this SW620 xenograft colorectal cancer model, where we subcutaneously injected that into nude mice. And then here we wanted to orally administer milk EVs and look at the primary tumor burden and see if milk EVs have any role, right? And again, we are orally administering milk EVs here just to mimic someone drinking milk. So in this case, our hypothesis was milk EVs may aid in cancer progression, right? Why? Because we are the only mammal who are drinking milk even after childbirth, right? Again, most of the organisms, like if you look at it, all the mammals, they give milk until for neonatal development, and after a while, the milk feeding stops, right? But however, we human being, we can do it, and we keep on doing throughout our life. So we thought maybe they can have repercussions in terms of growth of the tumor. So hypothesis was milk kiwis is going to aid in cancer progression. But however, it was opposite to our hypothesis, where you can see the PBS alone data here. This is the tumor volume, but moment we gave raw milk and commercial milk kiwis, the tumor burden, which is like 25 mg per kg to this mice, you can see the tumor burden was significantly reduced. And this effect was also observed when we looked at whether these EVs are reaching the tumor, and that you can see here, the EVs are indeed reaching the tumor, and this could be a direct effect as well, that we can see it's reaching the tumor. We also wanted to check this on a different model, a 41 breast cancer model. This is syngeneic models, which is developed in BALBC. These are immunocompetent models as well, as opposed to the previous one. We wanted to check whether this can happen as well as look at the role of whole milk as well as the EV component. So in this case, 41 breast cancer models were used, and then we orally fed milk EVs. And here the red bar, the PBS1, you can see the primary tumor is here. Moment you deplete the EVs from the whole milk, you can see the green one. There's no difference between the PBS and the milk depleted group. But however, when you give the commercial milk EVs to these mice, the primary tumor burden is significantly reduced, as well as when you give the whole milk to the mice. So this data suggests that milk kiwis are reducing the primary tumor burden. Also, in the whole milk kiwis are, whatever we sediment in that fraction, are the most important part of this phenotypic effect that we are seeing, right, where they're reducing the primary tumor burden. Even more interesting is, if you just treat the cancer cells, the 41 cells, with one dose of milk kiwis, either 200 or 100 micrograms per ml, and then take those cells and inject them into the mice, the primary tumor burden is still decreased. Just one dosage, collect the cells and put it in a way, suggesting that they're having that effect, anti-cancer effect in just one dose. So then the next question was, okay, we had a good effect in primary tumor. What about metastasis, right? Because again, metastasis is one of the main causes of cancer death. And again, we use this 41 syngeneic model, orthotopic model, where we implanted that on the memory fat pad, and then oral administration of milk kiwis was done. And then we wanted to look at primary tumor burden and lung met. And in this case, hypothesis was milk kiwis can attenuate or inhibit cancer metastasis, because we try to find drugs often in cancer. Very rarely do we get a drug that can work. In this case, milk kiwis were beneficial in reducing the primary tumor, and that's why we hypothesized that can inhibit cancer metastasis. Again, consistent with the previous data, we can see when we inject this to 41 breast cancer models, the primary tumor was consistently reduced. We used two dosages here, one the low, which is 25 mg per kg, as well as a high 50 mg per kg, just to make sure that if they can be curative, right? If they can reduce the primary tumor burden even more. But however, the primary tumor burden was not reduced, suggesting that there is a saturation here, right? Anything more than 25, it's the same, 50 also is the same reaction. However, to our surprise, right, when we gave these animals milk EVs, both raw milk and commercial milk EVs, despite the reduction in the primary tumor burden, it accelerated metastasis and killed the mice faster, right? Here you can see PBS alone. This is the lung flux, which is the bioluminescence. You can see the raw milk low, both of them have higher metastatic rate, as also with the commercial milk high group. You can also look at the relative tumor burden, which is nothing but qPCR for M-cherry, which is only present in the cancer cells. You can see here that all the groups have higher metastatic burden, especially the high groups compared to the PBS alone group. You can also look at the visible metastasis higher in the milk-treated group. All this data collectively said that milk kiwis is not good. So when I got this data, me and my student were looking at each other, and that was a Friday, and I was a bit surprised with this data. And then I swear I didn't touch milk for the next 48 hours. So then I was curious as well, what are we putting in milk kiwis? Why is this happening, right? So we went back to the drawing board, and then we sonicated the milk kiwis And then we wanted to see whether this effect can be completely abolished. And that's what we indeed saw, where the PBS alone didn't reduce the primary tumor burden as we expect, but the commercial milk EV reduced the primary tumor burden. But however, sonicated EVs did not have any effect, and it was similar to the PBS. Same way, when you look at the metastasis, the pro-metastatic effect of milk EVs was completely abolished when you can sonicate the milk EVs, as you can see here, suggesting that intact milk EVs is important for these EVs to be functional. So then we started asking the question in the clinic, in the patients, right? So what if the patient has the primary tumor surgically removed, and what happens to the metastatic burden? This is what you see normally in patients. So can patients who have metastatic tumor drink milk, right? So again, we are not testing the whole milk in every aspect here, but we still wanted to articulate this, whether milk EVs can have a different function, or they can accelerate metastasis in this context as well, or kill the patient faster. So this case, we again did 41 breast cancer model. We did a mammary fat pad injection, resected the primary tumor after 10 days. And at this stage, we already know the primary tumor has seeded onto the metastatic organs. And then oral administration of milk EVs was done, and we looked at lung metastasis. In this case, our hypothesis was nothing, because my track record again 0-2, and I was able to tell everything pretty much wrong, and my hypothesis was always wrong. And what it says is, like, if you have a problem, you can approach me and I can tell you a hypothesis. You can rule that out and work on something else, so that saves you time. And that's pretty much my track record here. But however, when you look at the data, right, so when you look at the surgically resected primary tumor, and then when you orally gave milk EVs to these guys, we can see this is the PBS alone group. When you gave milk EVs to these guys, you can see that it reduced the metastatic burden in all these mice compared to the PBS alone group. This is the lung flex, the bioluminescence. This is the visible metastasis, and this is the qPCR for M-Cherry, suggesting that surgical resection of primary tumor reverses the pro-metastatic effect of milk EVs, and it's beneficial to cancer patients, right? It may be beneficial for cancer patients. So the context in where you use milk EVs and the time when you administer milk EVs is very important. So we also checked other cancers like pancreatic cancer and human colorectal cancer, similar data where it accelerated milk EVs. We looked at cachexia in a way, but these are xenograft models where it reduced cachexia, but however, because these are xenograft models, the results were different, right? So we could not completely believe this, because if you use syngenic and orthotopic models, then the results are different, where milk EVs are accelerating metastasis. So what about the mechanism? When I looked into the literature, I couldn't find any study where they were showing about this contradictory effects, where the milk, or not the milk, any drug reducing the primary tumor burden but accelerating metastasis, right? So how is the milk EVs reducing the primary tumor burden, and how it is accelerating metastasis, and how this opposite effects are happening? So to cut the long story short, so we did MTS assay to see whether it has anti-proliferative effects, and it did indeed had anti-proliferative effects, but the effect was saturating, right? Similar to what we saw in vivo, we can see milk EVs have a saturating anti-proliferative effect. And when I saw this, I thought maybe it have some role in cell cycle arrest or senescence, right? But then we also didn't rule out apoptosis, right? So we looked at any caspase-based cell death was there. We used QBD and caspase 3-7 double knockouts, but there is no difference. We also looked for necroptosis using necrostatin inhibitor. There is no difference. We also looked for autophagy-dependent cell death using knockout cells and baphelomycin. In a way, there is no cell death in these aspects as well, suggesting that the anti-proliferative effect is mainly on the proliferation and nothing to do with cell death. Then we looked at cellular senescence, as I pointed out, because there was a saturation effect. When we looked at the P16 marker, the marker of cellular senescence, you can look at untreated cells do not have a P16, but the moment you treat these cells with commercial milk EVs, you can see P16 comes up, and that is quantified here, suggesting that milk EVs can induce cellular senescence in these cancer cells. Then we looked at the role of immune cells in the primary tumor, in the circulation, as well as in these metastatic sites as well. We could not see a significant difference in any of the immune cell subtype, nor in the primary tumor burden, nor in circulation. And that is like 3-4 figures, which is in the supplementary data, if anyone is interested in the published paper already. So we wanted to quickly see what are the pathways that are perturbed in these cells. So what we did is we took the commercial milk EVs, treated them with 41 cells, did a lysate and then proteomic analysis to see what pathways are changed. To cut the long story short, what we found out was, we found out epithelial mesenchymal transition pathway was changing, as well as cellular senescence that was changing in this proteomic analysis. So we validated that in the primary tumor by immunofluorescence. Again, EMT is epithelial mesenchymal transition, where the epithelial cells gain mesenchymal attribute and they can migrate. In this case, you can see PBS alone. There is proliferation. Chi67 is a proliferation marker. Vimentin is less, which is a mesenchymal marker. But the moment you treat with commercial milk and raw milk EVs, you can see the proliferation is reduced, as well as Vimentin signature is increased, suggesting that cells go into a mesenchymal phenotype. And this is also validated in the primary tumor by Western blotting. You can see Vimentin goes up, P16 goes up, and that is quantified here, suggesting that milk EVs is reducing senescence and EMT in the primary tumor. So this is the summary in terms of the story. And again, milk EVs is a double-edged sword. Normally, when we have a cancer, again, through epithelial mesenchymal transition, the cells can enter circulation and then can go and seed into secondary organs, thereby creating metastasis. But in this condition, when we give milk EVs and when the primary tumor is intact, the milk EVs is inducing senescence in the primary tumor, but however, accelerating metastasis by inducing EMT in these cancer cells, and thereby accelerating metastasis in the secondary sites as well. But when you resect the primary tumor, these EVs go to the secondary site where the cancer is already seeded. It is inducing senescence in the secondary sites and thereby reducing their growth. So they're not stopping the primary tumor migration, rather, because it's already resected, they're going to the secondary tumor and inducing senescence there, and thereby allowing them to not grow as much and providing a benefit. Anyway, so I would probably emphasize some caution as well here, because milk EVs are high. EVs are high in milk, but this concept cannot be attributed to every food source. And use of xenograft model, I would say a caution, because if I just use xenograft model, our whole title would be different, saying that milk EVs is really good for cancer, right? But however, that's why it's important to use orthotopic models to see what you get out of it. Lastly, I'll finish with this slide, because the reviewers, when we submitted to this Nature Cell Science journals, they asked us, okay, can all the EVs survive the gut, right? Why are you looking at milk EVs? Can you also put cancer EVs into the gut, right? And our hypothesis was, what is the relevance of cancer EVs? Why would someone drink that much amount of cancer EVs or eat that much amount of cancer EVs into the gut? So we thought we'll use this for other purposes, and that's why I rebutted to the editor, and editor didn't ask us to provide any data. But however, for curiosity, we looked at another beverage like milk. So when you look at milk EVs that is high in milk, the EVs are high in milk, but other food sources like melon, citrus, where when you break up the cells, they can form these EV-like structures, as well as there's some EVs also present in them. There may be more in them, but we didn't want to use cancer EVs because there's no physiological relevance there, unless and until you're talking about red meat, and then if some of them may have an impact later on. But we took beer, because beer is also a highly consumed beverage, and it's also a fun experiment to do, right? So you take beer, collect the EVs, and then when we gave this to mice, again, this is yeast EVs where we gave 25 mg per kg. They are able to survive the gut and reach other organs. But when you give beer, which is like 2 standard drinks, which is normally consumed in Australia, which is like 2 standard drinks, there's no, none of these EVs are able to survive, because there's very low amount of EVs in beer, right? As you can see here. So if you want to have this amount, which is like 25 mg per kg of yeast EVs in beer, you need to consume almost 7,000 beers a day, which is not physiologically possible for anybody. I'm not sure it's possible. That's why we just rebutted saying that the milk EVs are high in milk, but not for other things. With that, I'll stop. I'll come to the most important slide. Again, the work was done primarily by Monisha Samuel, who postdoc, who is a student in was a student in my lab, now postdocing in Karolinska, and then Rahul, who just finished and he went to UK for his postdoc as well, and all these wonderful collaborators. And then thank you all for your interest and listening to the talk. Thank you.
Thank you, Suresh, for the very mind-blowing presentation. And now the floor is open for the questions.
Thanks for the nice talk, sorry. So can you remind us again, how are you achieving this sort of dual effect that is on the primary tumor versus the metastasis? So you argue that on the primary tumor, it has a different function, whereas a metastasis is different than what's going on in the primary. So I'm still trying to understand how you can have this dual function from the same pool.
So in the primary tumor, the milk EVs are inducing senescence and EMT. So from the primary tumor, the cancers then try to migrate, and that's where when it's induced with EMT, they're trying to move from that primary site and go to the secondary organs, and that's what you see. But however, when you remove the primary tumor, the cancer cells, the EVs are then going to the secondary site and inducing senescence alone in those things. Because even if it induces EMT, we have not heard of secondary tumors migrating or metastasizing again, right? That is pretty much the concept that we are going by, because they're already settled in the organ. But now the concept of EMT is like when the cells going through epithelial to mesenchymal, they go to the site and they stay in the epithelial phenotype is what people kind of believe. And again, it's controversial. But however, in this context, we think the senescence is what is caused in both primary and secondary. But however, there's no EMT happening in the secondary.
Thanks.
Thank you so much for the wonderful talk. I'm going to ask a question that I'm not, maybe other people might know, but you said that EVs affect the physiology of the receptor cells. So I'm wondering, now that you have focused on EVs going to the primary tumor cells, what about if a mice that has tumor, and this tumor produces the EVs, and then I don't know if if you have milk from a mouse, from something that is producing a cancerous animal, then it produces milk, and then you feed this milk to a non-cancer animal. Do you think there's some kind of transfer of the EVs that are cancer to the non-cancerous animals and then maybe induce the cancer state?
I don't think so, because this question can be asked in a different way. So does consumption of milk or milk EVs cause cancer? I don't think so, but it can accelerate tumor which is already there, right? If you have a cancer in a way, then it can aid in acceleration. But even if you take from an organism that has cancer and then milk from that, I don't think it would have an effect. So we also looked at same species in a way, looking at mice to mice. We didn't see an effect. So to answer this question as well, so here we also looked at many other species like cow, buffalo, goat, sheep, and then all of these have the same effect, like in terms of cow milk. Every milk that we use, because 99% of the world population consumes any one of these milk, all of those have the same effect in terms of accelerating metastasis. But however, for your question, I don't think it can happen, but I don't think we have tested it, but my gut feeling is it may not happen.
Thank you very much for the nice talk, Suresh. Just one quick question. So you found that now the milk EVs can induce the EMT. When you look at the protein cargo, what were the candidate like that you found that would known EMT inducers? Did you find anything in your proteomic data set?
So yeah, this was the question asked by everyone, asked by the editors, the reviewers, everywhere. So wherever I go, the Nature Cell journals. So we spent 3-4 years chasing about it, because as I said, 2012 is when we started this. We published only in 2021. So it took us that many years to finish the story, because primarily we were chasing a cargo protein or an RNA. But however, this effect could be collective in a way, but we looked at TGF beta, which is present in milk EVs. TGF beta is known to induce EMT, but however, in our hands, we could not see that. When we used TGF beta from milk EVs by itself, it was not having the same effect. Lactocadherin was another one we chased, it didn't have an effect. So we chose somewhere like 10 candidates, but none of them worked in our hands. So what we ended up doing is we only proposed this as a mechanism and then got it out. So it was reviewed by other big journals, and then they asked this one question everywhere. So we were able to address everything except this one. So I had to pull out from there and then go to NatureCom for that reason. So we do not know is the answer. In other projects, we are trying to find out, but this project, we do not know.
Have you tried, except giving the EVs to the mice, the cow EVs to the mice, have you tried to give the cow milk EVs to any other animals?
No. So yeah. Do I count as an animal? So I drink, but I don't have cancer. Sorry. So we haven't tried that. I was curious. I was talking to some people who can do these kind of experiments. Like I was even looking at the same species, right? So like cow to cow, or even in terms of mice to mice. I was trying to set up some collaborations with that, but yeah, it didn't work out. But I was curious. So answer is I didn't try.
There are over nearly 300 papers on milk EVs, but nobody has done anything except putting it in the mice, or maybe there are 1 or 2 into the rat. And it's a bit, the field is very, how can I say, very limited if every experiment or all the information you are getting is coming from just one species.
That's right. So I think that's the problem with whole biology in general. But if you look at other organisms, right? Like I see some people using chimpanzees for EVs, and very few, like only 1 or 2 studies out there. Like there's also my collaborator was suggesting to use for horse as well. We were trying to look at EVs and using there from vein versus arteries. But again, these are the ethics. The running cost of all of them is humongous. People using zebrafish, yes, we have done in zebrafish, but not milk EVs. But yeah, you're right. So a whole biological field is somehow only looking at mice and rat.
If there's no more questions, okay.
I know someone asked a question related to this a little earlier, but so if you think about it, just mother to the progenies, right? So mother that are breastfeeding and looking at that effect there. So if you look globally in cultures where women breastfeeds more regularly, or it's, they're more religious about it, for lack of a better word, compared to others? I mean, do you have different prevalence in terms of metastasis and therefore death?
There's no epidemiological studies on that as well. Not even milk consumption generally. There are quite a few epidemiological studies, but however, it's contradictory. Some people say it increases, like say pancreatic cancer. It's beneficial for colorectal cancer. But however, in the context when they already have cancer, nobody has studied. It's more on the risk part. Even the same for mother to infant as well. There is not much on cancer per se, but rather the benefits of feeding. Probably there's a lot of epidemiological studies. There is indeed a lot of cargo from milk kiwis being transferred from mother to infant. And that has been also studied by several other groups as well. That is true, but not in the context of cancer. But again, there's a concept of breastfeeding. Again, when you're feeding milk, there's also high risk of breast cancer. In that case, whether any EVs have any role in that, I don't think anyone has studied generally.
So thank you very much. And I would like to say once again, thank you very much, Professor Suresh, for your mind-blowing talk. And we can go for the coffee break now. And I would invite you to walk around. We have quite several poster presentations, get more knowledge, exchange knowledge, and take this opportunity to communicate with your researchers here. For today, we'll take 30 minutes of break instead of 20 minutes, and we can come back at 11:30. Yeah. Hello, and welcome back to the second session of the day. Before going to the next talk, I have to give you a quite small reminder. We'll go for an extended lunch today. Just after your lunch, we would like to come near to the poster presentation area, and each of our poster presenters, they'll give a 3-minute talk about their poster, and then you can ask questions and give comments, and then can give directions to improve their work. I hope it'll be a good chance for the poster presenters as well. So next speaker, or the first speaker for this session is Johanna. Gandhi will give the speaker introduction.
So our next speaker will be Johanna P. Bor. She's a graduate from veterinary medicine in Estonian University of Life Sciences. At the moment, she is doing her PhD in molecular studies of subclinical endometriosis. Her work aims to characterize molecular changes in uterine tissue of cattle with subclinical endometriosis and its potential use in diagnostics. So today her talk will be on bovine uterine fluid extracellular vesicles, all the secrets for deciphering the pathogenesis of subclinical and clinical endometriosis. Johanna, the floor is yours.
Thank you for your kind introduction, Kayande. I will give you a bit more different presentation than maybe some of other people, because I'm a clinician and I'm interested in how this subclinical and clinical endometritis develops. And we hope that the bovine uterine fluid extracellular vesicles might show some kind of deciphering ways to understand this disease development. But briefly, first, that cow reproduction can pose various risks to the health of cows due to physiological demands of pregnancy, calving, and lactation. This reproductive cycle typically begins with either artificial insemination or embryo transfer to the future mother, where a fertilized embryo has the potential to implant around the 19th day of the estrous cycle. By day 35, cows undergo pregnancy confirmation. If the cow is not pregnant, the cow will go back to estrus and this cycle will repeat again. However, if the pregnancy is confirmed, the cow will calve approximately 280 to 285 days later. This process can significantly impact the uterus, causing damage to the superficial layers of the uterus, have bacterial contamination originating from the environment. And of course, there is an inflammatory response to these pathogens. While many cows can adapt to these changes and return to the estrous cycle, many may develop uterine inflammation, compromising their health, welfare, and fertility. One of the most prevalent uterine inflammations in bovine is endometritis, characterized by inflammation of the endometrium occurring 22 days postpartum. There are two distinct subcategories of endometritis. There is this clinical endometritis and subclinical endometritis. Some researchers have agreed that these two forms can independently develop, while others believe that subclinical endometritis is the continuation form of the clinical endometritis. However, we all agree that clinical endometritis presents visible signs such as mucopurulent pus discharge from the vagina, while subclinical endometritis doesn't have any apparent symptoms. However, they do have these polymorphic nuclear neutrophils in the uterus, which you can see in the picture. And therefore, this diagnosis of the subclinical form of endometritis at farm settings is relatively difficult. The primary diagnostic approach then is the proportion of polymorphic nuclear neutrophils, which is always higher compared to the healthy cows. But this proportion really depends on when the samples are taken and can have many different factors that can influence the proportion that can be detected via cytology. Therefore, better diagnostic approaches are really needed in farms. As a consequence of this inflammatory response, cows may develop infertility and ultimately be calved. In Estonia, approximately 13.8 to 20.5 cows are calved due to reproductive problems, ranking as the second most common reason for calving in the country. To really understand these diseases, we have to really understand what will happen in the uterine microenvironment. This microenvironment consists of freely circulating molecules within the uterus, as well as extracellular vesicles, so-called EVs, that can originate from different sources, just as immune cells, in endometrial cells, as well as from the microbiota, as we know that the uterus of cows is never a sterile environment. It's important to note that these EVs from mammalian origin as well as the non-mammalian sources cannot yet be reliably distinguished. Therefore, when we enrich the EVs from the uterus, all sources of EVs are isolated together. In my research, my main focus is primarily on the protein component of these EVs. Protein offer an alternative avenue for biomarkers because they provide real-time insights into the state of cells under specific conditions, showcasing rapid changes within the short time frame. These changes can be harnessed to develop potential diagnostic options or for subclinical endometritis by making an assay. These proteins on the EVs can be bound to the cargo or into the lipid bilayer, or they can be in the protein corona. In my research, I have done a few experiments on uterine fluid samples that are acquired from cows around 36 days postpartum who had clinical endometritis, those who had subclinical endometritis, and those who have healthy. And for purification of uterine fluid EVs, we use differential centrifugation coupled with size exclusion chromatography. Afterwards, these EVs were subjected to nanoparticle tracking analysis, where we could see that the concentration of particles in the clinical endometritis is significantly higher compared to the healthy cows, as well as those with subclinical endometritis. While going to the particle size measurements, we could see the opposite, that the clinical endometritis average particle size was significantly lower compared to those which were acquired from the healthy cows and those acquired from subclinical endometritis. However, the average particle size was significantly higher in cows with subclinical endometritis compared to those in healthy, where we couldn't see any concentration differences between these groups. This potentially may suggest that the ratio between the EV concentration and the size could potentially serve as an indicator for disease presence. In the proteomic analysis, we did leukochromatography mass spectrometry, and we saw a total of 1,549 proteins, from which 248 were significantly differentially enriched between the different groups. In the principal component analysis, it relieved that these groups of cows are the enrichment patterns are significantly different. However, there was a notable variability within the subclinical endometritis samples, characterized by a median coefficient of variation of 65%. In this heat map, we can see discernible differences in the differential uterine fluid proteins from clinical endometritis compared to those who were healthy and were with subclinical endometritis. Notably, the second cluster of proteins displayed depletion in clinical endometritis, while it was enriched in other groups. These proteins were mainly related to gene expression, regulation of translation, cellular nitrogen compound metabolic processes, plasma membrane organization, regulation of cell shape. The first cluster of the heat map, however, displayed increased enrichment of proteins in both clinical and subclinical endometritis, which were primarily associated with cell apoptosis, cell migration, and immune-related processes. Then we went directly to research immune-related pathways, as we could see that in these proteins were differentially enriched between these two diseases. And we could see that there are 15 immune-related pathways were consistently identified in both of the diseases. However, in clinical endometritis, there were 4 pathways that were unique, and in subclinical endometritis, there were 20. These differentially enriched pathways were linked to cell damage, apoptosis, and active inflammation aimed at eliminating pathogens in clinical endometritis. While in subclinical, the pathways showed regulation of aberrant inflammation process, including cell damage, active responses to fungal and bacterial infection at the same time as there was occurring endometrial repair. So we could state that these inflammatory responses is different between the two diseases. Traditionally, it has been held that during peripartum period, inflammation development involves the recruitment of leukocytes, which can help eliminate infection agents before undergoing programmed cell death. Nevertheless, the failure of apoptosis, the incapacity to counteract bacterial harm, and the persistent activation of pro-inflammatory pathways may play a role in chronic inflammation. We can see that when we compare subclinical and clinical endometritis, there are different responses to apoptosis, which was more prevalent in clinical cases, and leukocyte proliferation and endometrial repair, which was seen in subclinical endometritis, indicating that these disease developments are potentially totally different. Therefore, these diseases, our research shows that diseases are probably independently developed. In summary, the particle concentrations are significantly higher between healthy and clinical endometritis uterine fluid EVs, however, not between healthy and subclinical uterine fluid EVs. Proteomic profile between the groups are significantly different, as well as there is distinct expression patterns indicating various immune response and the separate disease mechanism in clinical endometritis and subclinical endometritis. Thank you, and I'm ready to answer your questions.
Thank you, Johanna, for the talk. And now the floor is open for the questions. Seems no questions for you, Hannah. So thank you very much. And then we have a token of appreciation for you too.
The Chief Executive Officer of Dispatec, he will be presenting about the technology behind determining the concentrations and analyzing the subpopulations of nanoparticle using nanocon technology. The floor is yours.
Hello, everyone, and welcome. Thanks for the patience. My name is Floris van der Staaij, and before I would like to start, I would like to thank the organizers for organizing this really nice conference. I think it's really nice to see all these people together, and I'm excited to see what's going out of all this nice research. So actually, I'm here to present to you a little bit more about the new device that we're developing. It's called the NanoQuant, and it helps with the quantification of fluorescently labeled nanoparticles. So we are a young and growing company based in Amsterdam, the Netherlands. We receive both private and public funding. And actually on the top right, you see the building, that's us, on the science park next to the Amsterdam University. And of course, we can't do this alone. So we work together with a couple of really nice parties, medical centers and parties that work on the development of new staining technologies and EV standards. So this is the NanoQuant. It helps with the concentration determination of fluorescently labeled nanoparticles. And besides that, it helps with the identification and quantification of subpopulations by the use of colocalization. And actually for every channel that you would like to use to obtain colocalization, you have the option to choose between 4 laser light colors and 6 filters, soon 12. The measurements are really fast. It's 30 seconds to 1 minute per channel. And we are now working on a new part of our technology, which would make it possible to measure up to 500,000 particles in one channel or in one measurement. Actually, at the moment, we have a concentration range of 3 orders of magnitude. And we are as well, of course, working on broadening that range. So after you've fluorescently labeled your nanoparticles, you are ready to use our machine. So what we actually do is we immobilize the nanoparticles in a hydrogel. And by doing so, this gives us the benefit that we stop Brownian motion. And therefore, we have the opportunity to illuminate the same slice of the hydrogel with multiple laser lights, which then are the channels that I earlier spoke about. And after we've done that, we use an algorithm to count the excited nanoparticles in that image that we've created. So it's really easy to make this hydrogel. It takes around 5 minutes. It can be done in batches before you start the measurement. So it's actually from after you've stained your particles, it takes around 5 minutes. You pipette your sample and 2 components to make the hydrogel, wait 5 minutes and you're ready to go to put it in the machine and it can be analyzed. So we don't illuminate one slice of the hydrogel, but we illuminate the whole volume. So what we do, we actually make a Z-stack of images moving through the volume. And after we've done so, we know very well the volume that we've just illuminated. And we also know very well the particles that are on the inside because we use a nice algorithm to count the particles. And with that, we can reconstruct back what the concentration is in that volume. So in a little bit more detail, how does it work? Here you see a top view of the hydrogel. And in this example, I'll show 4 channels. So use the laser light to illuminate one slice of the hydrogel. This creates this image, which can be seen on the left side in the hydrogel. After that, we want to maybe choose to change to the next channel with the use of another filter wheel. For channel 3, we might decide that we change laser color and filter wheel. And for channel 4, we again change the filter wheel. After that has been done, we use a very precise piezo device to move with up to nanometer precision. The hydrogel, the filter wheel moves back to channel 1 and the whole process starts over again. So channel 1, 2, 3, and 4. And this whole process continues until we've eliminated the whole volume. So now you have one slice of the hydrogel that has been cut, and you would have, for instance, 4 channels. So you would have 4 pictures of that one slice with the nanoparticles that have been illuminated. So this is an example of how it would look if you would use the 4 laser light colors that we've integrated in the device, 405, 488, 561, and 640 nanometers. So for each channel, you have the choice to choose between these 4 laser colors and an emission filter. So you make sure that you only see the particles of interest or the biomarkers of interest. And now we can do the colocalization between these channels. And as an example, you can see that in the top left channel, there are, for some of these particles, no biomarkers present that react to the illuminated light, while the same particles in the other channels indicated by the red circle are present. So it's really easy to use. It's really fast. It takes around 5 minutes to do the sample preparation. You don't wash anything away. And we did a linear series analysis. We looked at tetraspec, which is a particle standard from Thermo Fisher. And you can see a very nice linear line between 1 × 10⁶ to 1 × 10⁹ particles per milliliter. And as you can see, for 640 nanometers, we are already there. For the other laser lights, we still need to work on making this linear line possible. But we of course want to also move to the lower end of the range, and of course to the higher end as well. Besides that, we also now looked at dual stained EVs in a collaboration with Hansa Biomed. For these particles, these were stained with both EGFP and M-cherry. This is an example of one slice. It's, in this case, the colors are actually inverted. So the black dots represent the nanoparticles, or at least the dyes that are present on the nanoparticles. And we could see that there were 3 populations. A population that was dual stained with both EGFP and M-cherry, and also, of course, a small population, or a population with EGFP and M-cherry not dual stained. And with that, I would like to say thank you for listening to my presentation. I would love to hear your opinion on what we've been building, and please tell me what you would like to see next, and then I'll make sure to put it on our technology roadmap without this big promise, but of course we would love to hear your opinion. So thanks.
Thank you very much. And just any questions?
How do you measure the size?
At the moment, we don't measure the size. We are thinking, of course, of ways to add that as well to the device. One way will be to maybe not make a hydrogel from the start and to analyze it by Brownian motion. But we, of course, want to also think of other ways to measure it.
You're speaking about nanoparticles and the colloidal stability. And your hydrogel should have different kind of ionic strength in there, and particularly different kind of even ions there. That would affect the zeta potential and colloidal stability of the nanoparticles. It would result in agglutination of them, any kind of shells that you would use or so. And depending on which kind of the nanoparticles that you have there, it would change. You show, for example, with one sample, it is like that, but are you not afraid that your hydrogel would change the factors that somehow affect the binding of these particles together?
Yeah, I think that's a good remark. It's something to further deep dive into. At the moment, we don't see any reason why it negatively influences the nanoparticles in a way that we can't at least still quantify them. We do see the EVs in concentration ranges that we would expect them. And as well, as an example, we mixed 2 populations with different colors of EVs, the same type of EVs, mixed them together, and then we didn't see that, for instance, all the spots were colocalized, which would indicate that they somehow get pushed together. So that's one example of how we try to further experiment with what the effect is of the hydrogel. And of course, there are multiple hydrogels that we can use, but we are a startup, and that's part of our, for sure, it's part of our roadmap, yeah, to further dive into that.
Yeah, so I think yesterday we had somebody else, I think it was Perker, the company that was here yesterday, I don't remember.
Particle metrics, maybe?
Yeah, something like that, yeah. So in terms of price, I know it's quite not delicate for me to ask this question, but I will because it's pragmatic. So what are we looking at here? I mean, you don't necessarily have to give me the right number, but how competitive are you with the current landscape? Because one of the major barriers, I would say, for the acquisition of these technologies, right? So some labs can get 10 of these. Some other labs, I have to get all the labs in the corridor on one floor to get together to buy this technology. So price toxicity is a major barrier.
I think it's a really good question. And I hope that I now say the good number so we can shake the hands after that. But no, without jokes. Our machine is at the moment priced at 100,000 euros.
Okay.
Thank you, Flores. Maybe I'm trying to understand the niche, no, let's say, for this instrumentation. There is, for sure, user-friendly instruments are necessary for this community, and that they would be, and all what could do this instrument much better could do, I guess, light sheet microscope. It's not too much expensive, no, in this comparison. But does it only need to look and quantify EVs, or is it possible on the same hydrogel to put some organoids, spheroids, and look EVs inside them?
I think it's a really good question, actually.
Like for more intended for like big structures, like
No, I understand what you mean. Indeed, this technology can, I think, also be used for bigger structures. We haven't tried so, but I think it can be really interesting to combine that. So I think it's good to maybe we can discuss it after the lectures or after the presentation as well. And I will be happy to hear your thoughts on it, actually, from everyone.
Because organosalsa is you use hydrogels, matrigel, to put them inside.
Yeah. But indeed, if the fluorescence is high enough, we will be able to see things inside of things. And I think this device is actually very sensitive. The benefit of stopping the Brownian motion actually is that you have the option to eliminate a very small, very dim particle for as long as you want to. You can make a bleaching curve. You can see how long it takes before the dye bleaches, and you can optimize and choose if you want to eliminate it for, I don't know how many milliseconds or seconds, I don't know, without losing the particle of interest out of your focus.
So no more questions, and thank you very much for the interesting talk. And we have a small session. Thank you very much. And now we can break for the lunch. And around 1:25, we can start the poster session.
Hello, welcome back. We are going to resume our last session, evening session, and we will start our talk with talk of Professor Bradley. And before he starts his speech, I will give him a little introduction. Bradley obtained his PhD from Cardiff University in 2019, and he is currently working as Assistant Professor of EV in Infectious Diseases at the Department of Clinical Medicine in Aarhus University, Denmark. His current research is mainly focused on host-immune modulation by EVs released from parasites. And the title of his talk will be Parasite Extracellular Vesicle Show Diverse Function in Their Host. Professor Bradley, the stage is yours.
Hello, and a little bit later than advertised, and apologies. It was my son's a better alarm clock than my telephone, it seems. But yeah, it's a little bit different. We're going to talk about helminths, so parasitic worms. Here we have some Ascaris sum that we commonly work with. These are the giant roundworms. They're quite large. So why worms? There's over 1.5 billion people infected worldwide still. We have very good drugs. You can see this on the graph. We have a 500 milligram mebendazole treatment that reduces the eggs per gram significantly, but there's no sterilizing immunity against helminths. So reinfection is rapid. So as soon as we stop giving the drugs, people become reinfected. And these infections are chronic, and the worms are very, very long-lived, to the point that this is the data from my PI. So he self-infected with whipworm. And we realized at some point that his worm was now 6 years old. And this was not known before. They assumed it was only 2 years. So these worms can now live at least 6 years. Some of the trematodes can live up to 20 years in a single host. So it's how they actually manage to do this, obviously avoiding sterilizing immunity. They're potent regulators of the immune response. There's an entire branch of the immune response dedicated to parasites, the Th2 response. The allergy and asthma researchers keep trying to steal it, but it is dedicated to parasite responses. They downregulate Th1, Th17 responses, and they upregulate this tolerant Th2 response. And we commonly see T-cell anergy and also expansion of Tregs in infected people and in animal models. The point of this is it enables the survival of the parasite, but it also enables the survival of the host, because they induce serious tissue damage when they move through the body, as exampled here by some great work by Judith Allen in Manchester. This is a hookworm, so it has a migratory stage where it will go through your lungs, eventually ending up in your intestine. And we can see the damage that's accumulated in the lungs fairly rapidly, but it restores quite quickly too. So what does it mean? Immunity is altered where helminths are endemic. So we see a much reduced incidence of autoimmune disease, such as MS, where helminths are endemic. Obviously, there could be many, many reasons for this, but we still like to use this slide. This is some work from a multiple sclerosis professor, Jorge Corrales. He had some patients that became accidentally infected with helminths. And as we can see, their remission was prolonged. And when they were actually treated to get rid of the helminth infections, they returned back to controls. There's been multiple trials, human trials with live infections since. It hasn't quite replicated the effects we see here, but they are ongoing. On the darker side, if you're infected with a helminth, you'll have a much reduced response to vaccines. This is commonly known, and we see 3.4-fold decreases in infected children compared to uninfected children. And quite interestingly, deworming, it is effective to some degree, but you need to start at least 37 days prior, and it needs to be multiple doses. So the effects are long-lived, and this has been confirmed in mice, where you can see quite an extensive deworming program was required to restore some of the vaccine efficiencies. So obviously, with it being an EV meeting, we were interested, are EVs involved in host parasite communication? So this is the 3 different worms we're going to introduce. So we have the Ascaris sum and the Trichuris suis. These are both nematodes of pigs, but they can and will infect humans. The Anisakis simplex is the sole reason that we freeze fish for sushi. It's quite a common accidental infection of humans. It causes quite significant pathology, and it's quite unique that it's the only parasite that's a registered food allergen. So in areas of Japan, more people are allergic to the actual worm than are allergic to fish itself. So we're going to introduce them as the good and the bad. So we'll start with the bad. It's quite a simple experiment for us. We can literally buy herring from the fish market. We isolate the worms from the body, and then we can just culture these for 3 to 4 days and isolate extracellular vesicles. So they do produce EVs, and we can see vesicular structures when we look using TEM or cryo-TEM and AFM. Because we don't have antibodies, obviously, to many of our worms, so we tend to use proteomics every time we do an isolation, and this is how we try and validate that we actually have EVs and that these are differential to other components of the worm. So we have the ES, which is a start material, the supernatant from the ultracentrifugation, and then the SP, which is literally worm homogenate, and we see good separation, and we see some proteins that we recognize as being EV proteins. So given the role of this worm in allergy, we wanted to see, are these EVs taken up by antigen-presenting cells? So we developed a new labeling method because helminths don't synthesize their own fatty acids. They take them from the host. So we used fluorescent lipid analogs, and we can actually feed the worms with these fluorescent lipids and then isolate EVs afterwards that are fluorescently labeled. When we perform these uptake studies, if we include serum from anisakis positive patients, as in with a sensitivity to it, we get an increased uptake, but we also see some increased uptake with the negative serum, which seems that there might be some non-specific effects as well of the IgG. Regards to the allergen, when we look at the different fractions, we see many of these registered allergens, mainly in the worm product itself. We see a few in the actual secretory products, and we only see a single one in the EVs. But when we've analyzed the proteomics, we see quite a lot of cross-reactive or potentially cross-reactive allergens, including to dust mites and then various collagens. And again, these are quite common EV proteins. So we have the NLAs here and the heat shock proteins. So we tested the EVs to see if they have any pro-inflammatory effects. So the first thing we did was we did a complement activation assay because C5a is a potent anaphylotoxin, and we know that a simplex infection can induce anaphylaxis. So when we've increased doses of EVs, we see an increased induction of C5a. We don't see the same for HEC293, and we don't see the same for worm homogenates or the actual starting secretory products. So we seem to think this is quite specific to the EVs of this worm. If we do this in the presence of cells and also in the presence of non-heating activated human serum, we can see that this augments the TNF production, but they are quite pro-inflammatory themselves. Again, from the allergy perspective, we wanted to see, is there something specific from the EVs that can induce allergy? And our overall aim is to find a particular allergy that we are cross-reacting with. So we started with house dust mite patients. We didn't see so much. Then we moved to polyallergic patients that had multiple allergies. And in those patients, when we do the basophil activation test, so we double gate on CD123, CD193, and coincidentally, CD63 is the activation marker of basophils. We can see that the EVs induce basophil activation. The ES, the worm homogenates, do not. And in the non-allergic controls, or to the best of our knowledge, non-allergic, we didn't see any response to the EVs. So in conclusion, the A simplex, it produces EVs. They're uptaken by antigen-presenting cells. They're pro-inflammatory, and they can mediate basophil activation in polyallergic individuals. Now we move on to the Ascaris sum. So this is the good, depending on your perspective. We isolate these from pigs from the slaughterhouse. It's as grim as it sounds. We culture extensively. We've had to optimize for a very long time with antibiotics, because if you can imagine, these are coming from the, you know, intestinal lumen of a pig. We've optimized fully now, and then we've isolated EVs. We've characterized in various different ways, but more importantly, we've looked at the functionality. We've looked at the microRNA content, just to see that there was actually specific differences between the supernatant and the isolated EVs. And we've done the same with the protein content. It's not on this slide, but there is a difference between the worms. They secrete some EVs, but they also have their own EV mechanism within themselves. And we've actually looked at differences between these two sort of populations of EVs, and we actually see differences between the ones that are destined for, you know, interactions with the host, and those that are actually mediating sort of worm, you know, natural behavior. We went to a very simple LPS-induced inflammatory model just in PBMCs, and we could see that these EVs, in contrast to the Anisakis ones, were actually anti-inflammatory. We know from infection in pigs, if we take the pig plasma, we can see that they're immunoreactive to the EVs. So we assume that even though the worm is living in the intestine, the EVs are in contact with the host to a degree that we see immunity against the EVs. So then we did a very similar study to what we saw yesterday. We wanted to see what target cells the EVs might hit. So we incubated PBMCs with the pre-labeled EVs again, and we saw predominantly CD14 monocytes with the recipient cells. And we confirmed this by imaging flow cytometry, just to make sure. What we did notice was, and this was quite a rapid process, that the EVs from Ascaris can change the profile of the PBMCs, or particularly monocytes. So we start to see a reduction in CD16. Because it's quite common in the worm field that glycans mediate some of these effects. And we noticed that the Ascaris EVs are heavily glycosylated. I mean, you can quite clearly-ish see it on the cryo-TEM here. And here on AFM, you can see when we have the native EVs, you can see the sort of periphery. And then when we PNGase-F treats to remove the glycans, you can see that this is completely gone. So we assumed that by removing these glycans, we might affect the functionality of these EVs. But we didn't. It showed no changes in uptake. It showed no changes in the functional effects. So we can conclude that it's not due to the N-linked glycans, at least, and perhaps these are just mediating stability for the parasite EVs. We did see, because it was such a rapid response, we assumed it might be a protease that's mediating this effect. So we incubated with a fairly broad spectrum metalloprotease inhibitor, and we could see that it's partially rescued when we have the inhibitor present. So we believe that some of it is dependent on a metalloprotease. We can't determine whether this is a protease on the EVs or whether this is actually within the cells. So for the functional effects, we know, as I've mentioned, that we get T-cell anergy when you have a Helminth infection, but we also show that the EVs target monocytes specifically. So we looked at the monocytes, and we could see a massive increase in PDL1, the checkpoint. And I'll just bring your attention, this is EVs per cell. And we see an effect, this is a very, very low dose. I mean, 3,500 EVs per cell. I think maybe 25,000 EVs corresponds to about 5 micrograms per mil if you're looking at protein. So this is quite a low dose to be seeing an effect. And obviously this is particle number. As we know, that probably doesn't mean EVs, so it could be even lower than this. What we do see is that when we treat the PBMCs with Ascaris EVs, we get a decrease in interferon gamma production when we do polyclonal stimulation of the T cells. If we deplete the monocyte population, this effect is lost, and in fact, it goes the opposite way. So in conclusion, we show that Ascaris EVs are anti-inflammatory. They target human monocytes, so they induce a novel phenotype with CD16 negative, PD-L1 positive, and also CCR2 is reduced. And they affect T cells, but it's specifically via monocytes. This is very, very recent. We've not done the histology yet, but this is a colitis study in mice. And we can see that it points towards that the Ascaris EVs, when administered IP, are protective in this colitis model of DSS. Interestingly, they were not protective in TMBS. So we need to dig a bit more down into the immunology to try and see why that is. But it's very promising to date. The last worm we talk about is the Trichuris suis. So this is the whipworm. It's very similar to the human whipworm, Trichuris trichuria. This is actually the worm that my PI has. It resides in your colon or the cecum, and it has a very interesting sort of infection niche in that you can't see it here, but there's a thinner portion of the worm called a stichosome, and that actually burrows into your tissue, and it forms a syncytial tube of cells. We don't know how yet, but this has been trialed many, many times in MS and colitis in human studies. So we did the standard characterization. We isolated by SEC, looking at the protein and particle ratios. We used AFM to confirm we had vesicles. Again, because we have no antibodies, we have to rely on proteomics. So we look for, intrinsic component of membrane was enriched in the EVs, but not in the other fractions. And we identify proteins that we now know are Helminth EV proteins. In contrast to the Ascaris, we saw zero effect of the EVs in our basic LPS model, but we do see an effect from the EV depleted fractions, which makes sense to us because we know that this particular worm secretes massive quantities of PGE2, and that most of its effects on the immune system are mediated by PGE2. So we'd spent a lot of time, money, and effort to get to the point where we had no immune modulation, and we needed to find something. So we focused on the microRNA analysis of the worms. We compared free microRNAs with EV microRNAs, and we also did the same for L1, which is the infective larvae. And we could see significant differences between the microRNA populations. And as we dug sort of deeper down, these are two that we found quite interesting. So these showed significant homology to two human microRNAs, so MIR22 3P that was mentioned earlier on, and MIR29A 3P also. And we can see in the all-important seed region, it's heavily conserved between the worm microRNAs and that of the humans. These two microRNAs are known to be downregulated in colon cancer. So this might seem like a bit of a stretch, but there's 3 helminths that are actually class 1 carcinogens. If you have schistosoma hematobium, for long enough, you will get bladder cancer. But there's no association of gastrointestinal helminths with cancer, despite the fact that they induce massive epithelial cell hyperplasia, tissue repair protocols, and this dampening of host immune responses, Treg cells, M2 macrophages. And again, it's just an association, but colorectal cancer has been historically low in helminth and dericaris, but it is rising. And these most significant rises, if we look at Japan, where they've introduced Ascaris control, we see a huge increase in the rates of colon cancer. Whereas uniquely for Japan and Korea, they've not adopted the American diet so heavily. So perhaps there is a role of helminths, perhaps not. So we wanted to see if these microRNAs could be functional. So we synthesized the microRNAs and we just simply transfected them into SW480 colon cancer cells. And then we used quite a robust assay. We focused on proteomics, because obviously, if it's not fully complementary binding of the microRNA, you don't get degradation of the transcript. So we prefer to look at protein, so then we know what is downregulated is really downregulated. And we did that at 48 and 72 hours, and we had obviously a scrambled control. So when we've done this, importantly for us, we saw many targets that were downregulated that are experimentally validated targets of the human microRNAs. So we're more certain now that these microRNAs might function just like the human microRNAs. And then we've done just in silico binding analysis just to see if the seed region can actually bind in the UTR. And it seems like it does. We've redone the analysis now based on isoforms because there is some differences in splice variants, particularly in cancer, and that's actually improved the data. It's got a better resolution now, even though there's sort of less movement on the expression. And we saw it doesn't mean so much, but we obviously looked at the pathway enrichment for these downregulated proteins, and we did see quite a lot of pathways implicated in cancer, but majority in metabolism. When we've looked at the cancer genome database, this is just 3 randomly selected, but this follows a pattern for most. We see that these are actually downregulated in colon cancer. So in conclusion, for Trichuris suis, we have microRNAs with a homology to human microRNAs that are reduced in colon cancer, and these are specifically enriched in the EVs. They downregulate proteins that we know are targets of the human microRNAs. We still need to find out if they actually have an effect. But it's a little bit tricky with the colon cancer. We've not quite found a model that we could use at the moment, but we're still working on it. So in conclusions, parasitic nematodes release EVs. They have differential cargo, which I suppose hints towards specificity. And these effects are on the host are dependent on the species. It must be remembered that Anisakis, the sort of allergic helminth that we looked at, that is not a natural parasite of humans. So we are an accidental host. It really wants to be in a cetacean, so a dolphin or a whale. And there seems to be really strong species-specific effects within helminths. This is maybe the first indication that EVs might be mediating some of this specificity, but we need to do much more work. And it also might be the result of the infection niche. As we showed, the Trichuris lives within your tissue. It's in direct contact with the host. Maybe it has less of a need for EV communication than, for example, Ascaris, which is it's sort of free living in the lumen of your intestine. I'd like to say thank you to the NISOM lab. So here we have the current members and some past members and some of our collaborators, particularly the EV Foundry. Of course, thank you to the pigs. Without those, we wouldn't have any EVs to work with. And thank you. And thank you for the invitation. It was fantastic.
Thank you very much for the interesting talk. Now the floor is open for questions.
Thank you very much. It's fascinating to see how especially the immunomodulatory point of the pigs. One, out of curiosity, just the pigs in Denmark, do they all carry helminths with themselves? Do they all have the
only the organic ones?
The organic ones. Then
Yeah.
that is all. And then one thing that strikes me is you do in vitro culture of these pigs. If I'm not the pigs, the worms that you are saying. But in reality, in the time that they are in vivo, they are constantly in a communication interaction with the host, that is the intestinal cells. Is it possible that the EVs that you are somehow working with them is actually different from the EVs that they are producing when they are in the intestine? Is it not better to have your system maybe in situ culture of the worms and the
Yeah.
intestines?
I mean, experimentally, it's tricky to do that. I think anytime you culture something outside of where it should be, it's going to change to a certain degree. I mean, we try and back up what we do with live infection studies. So if we pin an effect to an EV, we try and look back at some of the live infections we've done and see if we see a similar effect. But it is a bit reductionist in that way, but it's the only way we can really sort of culture and get enough EVs for this.
Majority of your immunomodulatory work is the standard ones that is use monocytes or the other blood cells. How is the situation if you would use intestinal epithelial cells to see how they react?
I mean, we are working on organoids. If I just go back, I mean, parasites, it seems like quite a niche area, but if we look here, I mean, the tough cells that are resident and the ILC2s, I mean, these were literally discovered by parasitologists, and it's a massive part of our immune response. So it isn't actually an immune cell that first responds to a parasite. It's, like you say, a enterocyte or one of the tough cells. So I mean, organoids is definitely the way forward. Thanks.
Thank you so much for the interesting talk. So yesterday we were discussing about the specificity, and then this discussion, I think it points to the specificity of the EVs towards, I think you said they're specific to the monocytes.
Yeah.
So what is the mode of like interaction? How are they entering the monocyte, and why are they specific to the monocyte?
We have no idea, to be honest. We assume it's not fusion, because then we'd expect some T cells, B cells to probably pick up as well. One thing we have tried to do is inhibit by sort of increasing incubation of non-labeled EVs. So the thought was that if we add enough non-labeled EVs, it should saturate the system, and we start to see a decrease in the uptake of the fluorescent EVs. We never reached that point. It doesn't seem to saturate. We've shown some inhibitors of phagocytosis can stop the uptake, but that's about as far as we've gone.
So is it phagocytosis?
Possibly. I mean, the inhibitor we used, it's not super specific, but I mean that's our best guess at the moment.
So I'm not trying to wake up the conversation from yesterday. I'll let it. I'll let it go. But assuming that this is not mediated via like lipid sort of at the membrane, right, on the EV and on the macrophage. So have you tried just sort of shaving these EVs and then repeating the experiments? I think that will partly address.
Yeah. Let's try and find it. Yeah. So we like a really simple sort of PNGase F. So we've only removed N-linked glycans. So that there is some evidence that O-linked glycans might be important for worms as well. So we should really repeat with the treatment to get rid of the O-linked glycans. But as regards N-linked glycans, they made no difference to what we were seeing. Yeah, we really thought that would have been, it would have done it for us, but it didn't, unfortunately.
Any more question? Okay, actually, I have one question. Like in the previous part, the Ascaris one, they have more remediation effect compared to the one later with trichoroid Swiss.
Yeah.
What do you think? Like, what is the reason why we don't see any effect?
I think it's because of where they live in the host. When we've worked with Trichorosuris before, like not in an EV based way, but just looking at what it secretes, and it's huge quantities of PG2. And almost every effect that we see by this worm in an immune response, we can replicate just using PG2. I think it's just proximity. It's easier for it.
Another thing about this worm secretion pathway, are they following what we see in normal conditions, or like
with the escort and things? Yeah. They have pretty much exactly the same machinery. It's very, very similar. One thing we don't know, I'm hoping to get a student looking at this. We don't know about the lipid pathway, so the ceramide dependent. That's something we'd like to look at and see if that's the case, because it's very odd with worms. I mean, they don't make their own fatty acids, so I mean, for them to produce EVs and secrete, it's a crazy thing, really.
Thank you. Yeah. If nobody don't have any question, yeah, we have a little gift to Professor Bradley. I forgot to announce one. We made some change to the program because some of our speakers didn't come from the early morning. So we will have a coffee break a little bit late, and we will have also short discussion or moderation, because I see most of you are look a little bit tired. So we will make it shorter. Yeah.
Thank you, Gidnit. With that, I would like to invite to our final speaker of the today conference, Dr. Mina Halapeni from the University of Helsinki. Mina completed her doctoral degree in physiology and neuroscience from the same university. And at the moment, she's working as a postdoctoral researcher at the lipidomic co-facility at the University of Helsinki, Finland. And today she will deliver her speech on lipidomics and metabolipidomics as powerful tools in EV research field. And over to you, Mina.
Yeah, thank you very much for the kind introduction and also for the kind invitation. I'm happy that you're all here and excited to hear about lipids and lipidomics. I'm sure it's very fascinating to all of you. So yes, nowadays I work as a postdoc at the University of Helsinki in the lipidomics core unit. I will just put the pointer here. So yes, my PI is Reijo Kakela, whom I worked with already during my PhD studies, and here's our team. And but the main part of the talk I'm gonna tell you today are from my PhD time. So when I worked with Finnish Red Cross Blood Service with Erja Kerkelä and Saara Laitinen, and also we did collaboration with Pia Siljander, and Sami Valkonen, who was a PhD student at the time, is also heavily involved in these studies. And so the lipidomics part I did in Helsinki, but for the lipid mediator part, at that time I went to the laboratory of Dr. Jesmond Dalli in London for 3 months to conduct the experiments there. Okay. Now about lipids. So I think what typically comes to mind when people think about lipids, well, is fat. So adipose tissue, which contains mainly like neutral storage lipids. What we also think about maybe when we think about lipids are the lipoprotein particles. So LDL and HDL that go around in circulation, they are involved in fatty liver disease and atherosclerosis, for example. And well, of course, when we think about EVs, the next thing that comes to mind is the membrane lipids. And the membranes consist of phospholipids, sphingolipids, cholesterol, glycosylated lipids. Yes, all very different, many different lipid classes. We also have these so-called signaling lipids that are also sound derived from phospholipids, these phosphatidylinositols that initiate the secondary signaling cascades, and also ceramides and lipid mediators that will be one of the focus of today's talk. But I do want to emphasize, so even though we have these lipids that are called signaling lipids, of course, they are very important in cellular signaling, it doesn't mean that the rest of the lipids are not signaling or active. So the lipids are not passive. So not just membrane structures or only functioning in storage. But one thing I hope you will get from all of this, that lipids are also active players in the membrane and also in the storage lipids. And so today we will focus more on the membrane lipids and also the lipid mediator part. And here you can see a cell membrane. I'm sorry that this graph is a very static, conservative way of depicting a lipid membrane. This is because, well, it's hard to make a figure that really would encapsulate the dynamic of a lipid membrane that is in constant motion, and the lipids and phospholipids are constantly moving, and also the fatty acids in the phospholipids are changed very often. And so the main phospholipid classes you can see here, they're phosphatidylcholine, phosphatidylethanolamine, phosphatidylserine, phosphatidylinositol, and sphingolipids. So yes, and they have an asymmetry in different leaflets. So yes, now we have the phospholipid here. So it has the head group, and it has two fatty acid tails. So it has fatty acids attached to it, and those fatty acids can be remodeled. So now there is this blue fatty acid here, but it can be cleaved away, for example, with a phospholipase A2, and then another fatty acid can be inserted into the membrane. And this is how the lipids are remodeled constantly. But also the fatty acids, so that are in the membrane, they can be again cleaved away with the phospholipases. And in the cytosol, for example, here, an example of docosahexaenoic acid, DHA, it can be enzymatically converted, for example, by lipoxygenases into a lipid mediator. For example, here, an example of resolving D6. And these lipid mediators are hormone-like molecules that are having effects on the cell of their origin or the neighboring cells typically. And also, for example, prostaglandins are part of these lipid mediators. So where, as I mentioned, these mediators are coming from fatty acids. And now, again, word that most of you have most likely heard of. So we have two very important fatty acid families. They are called the omega-6 and omega-3 fatty acid families. And the omega-6 or N6 fatty acid family, the main precursor for these lipid mediators is called the arachidonic acid. And it has 20 carbons and 4 double bonds, hence the name 20 and 4. And the N6 just means the place of the fatty acid family, so the place of the first double bond. And arachidonic acid is a precursor of mainly pro-inflammatory lipid mediators, prostaglandins, leukotrienes, and thromboxanes. But prostaglandins have a very weird role because they also can have anti-inflammatory effects. And arachidonic acid is also a precursor of pro-resolving mediators called lipoxins. And these N3 or omega-3 fatty acids, you have heard most likely the hype by these pills. Well, these you can you should get from the diet, so you can get them from fish or marine algae, where they are actually originally produced. And from EPA, you can get E-series resolvins, and actually also these prostaglandins and other mediators, but they are less pro-inflammatory than the ones that are coming from arachidonic acid. And DHA is a precursor of multiple other types of these lipid mediators. So yes, lots of new words. But what is the function of these mediators in the scope of inflammation? So first we have a trauma, injury, and initiation of the inflammation. So we have edema and swelling in the inflammatory sites, and this is mediated by the pro-inflammatory mediators. For example, here from the lipid mediator perspective, the prostaglandins and leukotrienes that also attract neutrophils to the inflammatory sites. And then when the neutrophils are peaking, then we have the acute inflammation. And actually, the acute inflammation is required in order for the lipid mediator class switching to happen, so that the production of the pro-resolving mediator is initiated. And then we have the reduced amounts of neutrophils in the inflammatory side by the recruitment of monocytes and macrophages. And finally, we achieve the resolution of inflammation, which is the active dampening of inflammation. So inflammation doesn't just passively dampen away, but it's actively regulated. And because these mediators are derived from fatty acids and from cell membrane, the cell membrane has a key importance in the inflammatory status. Because if we don't have the fatty acids on our membrane, then we can't produce these mediators. And of course, we also need the enzymes and everything, but also the fatty acids. So why lipidomics and EVs? Well, as you know, EVs consist of these bilayers, and lipids are very important in the signaling pathways between cells and EVs. And also membrane composition is altered in many disease states. And also EVs transport many lipid mediators involved in inflammation that I will also tell you later a bit more. Some characteristics of EV lipids. So the lipid profile of EVs resembles that of the cell where they are being produced. But there are also some specific things that are common for most EVs. Most likely some of them are due to the curvature of the EV. So it's very tight curvature when you compare it to a cell membrane, which is the curvature is not that tight, but more loose. So that's one of the reasons why the asymmetry has to be more tightly regulated, most likely. So there are some enrichments of certain lipid species, lipid classes in EVs, for example, cholesterol and sphingomyelins that are important in multiple lipid rafts, and glycosphingolipids and phosphatidylserines that are important in cellular communication. And EVs tend to contain, they don't have lipid droplets, so they have low storage lipid content, such as triglycerols and cholesterol esters. And well, how to then analyze these lipids? Well, typically lipidomics is done by mass spec. There is also NMR possible, but it's not as sensitive as mass spec. And as the EV samples tend to contain typically a low amount of material, a high sensitivity application is preferred. And of course, if you have only a little bit of sample, then you might not be able to characterize all the lipids in your sample. And coming to that, so what would be a good amount of EVs for the analysis? Well, it really depends on the cell type, where they are originating from, because different cells have different amounts of lipids, so it's hard to say for sure. And also the EV isolation method, of course, makes a difference. For lipidomics, it's easiest to pellet the sample, because then we get rid of the solvents, or the solution that the lipids are in. But of course, depending on how we purify the samples, how we collect them, of course, we might have like wrong lipids in the sample also. And a good idea is to like do a small pilot test, in my opinion, before starting to do a larger analysis. Well, what is really lipidomics and metabolipidomics? Horrible word. So lipidomics is the analysis of the lipidome and membranous storage lipids, and metabololipidome is the analysis of the metabolically active lipid mediators, to put short. And so I'm gonna present to you two studies, a study with mesenchymal stromal cells, where we analyze the lipidome of cells and EVs, and also the lipid mediators from a sample where we had, from a cell culture sample where we had the cells and the secretome, so the conditioned media all together, in order to make sure that we have enough material. Then there's also a study with platelet concentrates and their storage time. And there we analyze the lipids and lipid mediators from the whole concentrate, the platelets, the cells, and also the EVs. And mesenchymal stromal cells, so they are used in experimental treatment of many immunological disorders worldwide. So in cell therapy applications, they are immunomodulative and anti-inflammatory, and multiple of their mechanisms of action have been elucidated. For example, their effects are partly mediated by EVs, which is why the interest here. However, their mechanisms still remain incompletely understood, and also the response of different patients vary for this treatment. And there are studies that indicate that the membrane lipid composition has an effect on the immunomodulation of the MSCs. So we were thinking if we could see if we can change the lipid composition of the membrane, and maybe that way we can see any improvement. And we studied the bone marrow-derived human MSCs, and they were cultured on plates, and the EVs were collected from these two stacks via ultracentrifugation. But the research questions first in more detail. So we wanted to ask if the MSCs produce these pro-resolving lipid mediators. It was not known, and it was not known if this is part of their modulatory mechanisms. And typically when we culture cells in cell culture, we just put the FBS there, and it's devoid of these 20 plus carbon polyunsaturated fatty acids. So it's devoid of these arachidonic acid and EPA and DHA. So we wanted to give the cells these fatty acids and see if we can modify the membranes, and does it have an effect to the lipid mediator composition. And so we gave the cells these fatty acids. Here as an example, the DHA. And then from these modified cells, we looked at the phospholipid composition, fatty acids, and also lipid mediators. And first we did like a time scale study. We could see that already after 2 hours, there was some modifications of the cell membrane, but the most prominent effects to the cell membrane was achieved after 24 hours in the phosphatidyl-choline lipid profile. And when we compared the cells with the EVs, we could see changes, and I will now show you some examples. So here, for example, you can see is the lipid species 40 and 6, and this is the combined number of the two fatty acid chains in the lipid. So they're most likely 18 and 0 plus the DHA that was added or given to the cells, and this 40 and 7 is most likely 18 and 1, and with the DHA again. And we can see the same, if even a larger effect in the EV. So after giving the fatty acids to the cells, then for 24 hours, and then we wash away the fatty acids, and then we let it sit for 2 days, and then we isolated the vesicles, and we could really see that the vesicle membranes were also modified. And you might think, well, it looks small, so tiny, but you need to think that not all lipids or fatty acids are like equal. We can't have like this many DHAs on our cell membrane, because it's so like increases the fluidity of the membrane also, and then the membranes would just break. And yeah. And so yes, what we confirmed here, that we can modify the membranes of the cells and the EVs. We were very happy. Also, not showing the results here, but the fatty acid profile was modified, and also the signaling of the cells was modified. So when we collected the cells and the cell culture medium and analyzed it with the lipid meters, we could see that the mediators from the downstream pathways of these fatty acids were increased. And so this is just basically the recap of the same. And then we did a follow-up study where we looked at the immunomodulatory effects of these supplemented cells. We did see some minor changes with the DHA supplementation, but here we looked at like regulatory or tolerogenic macrophages. In retrospect, we maybe should have looked more naive monocytes, maybe would have seen different effects. But nonetheless, our idea is still that that with PUFA supplementation, that would be like very easy way to modulate and modify these cells, because I mean it's just you give them something, FPS or PRP, something to the cells to grow, and then you could just give these fatty acids. So it would be a very, very safe way to modify the cells. Then the next study was a platelet concentrate study. So these platelets that are given to into the clinics as transfusions, the maximum storage time of these is 5 days, because the platelets activate after that time. And the idea here was to look at what is the lipid and the lipid mediator composition of the platelet concentrate. The lipid composition had already been investigated by other groups also. And we also looked at the changes in the storage. And this time points investigated were day 1 to 5, and also at day 8, which then the product would already be an old one. And when looking at the lipid profile in general, so we could see that especially the whole concentrate and the platelets, all the samples from different time points grouped very much together. So the samples were quite similar, but when looking at the EV lipid composition, there was most variability due to storage. You can maybe see, or maybe not. So the day 1 and 2 group over here, and the day 5 and 8 are more in here. This, I believe, is partly because these concentrates also have lipoprotein. So in these first 2 days of these samples from the first 2 days of the EVs, then the cells haven't been able to produce that many EVs yet into the concentrate. So they have like bigger ratio of the lipoproteins also in the sample. But when they start to produce more and more platelet-derived EVs, then the ratio with the lipoproteins becomes like less. And so mainly here they are the lipids derived from the platelet vesicles, but here we also have some lipoproteins in it. And when we look at the lipid mediators, what we could see, because as the things will accumulate in the bag, they're not going anywhere. Also, the lipid mediators accumulate in the bag. And these are the the arachidonic acid derived, mainly pro-inflammatory mediators, thromboxane B2, very platelet specific, important in platelet function, and then prostaglandin D2 and PGE2, which we also heard in the previous presentation. And so all of these accumulates in the concentrates, and also we do see some accumulation in the EVs, but the only clear accumulation was with the PGE2 in the EVs. And well, we also saw that in the concentrates, they produce also lots and lots of these pro-resolving lipid mediators. For example, these are derived from DHA. There's a clear increase in many of them. And also, when we look at the EVs, we see some increase in the actual lipid mediators, but we also see an increase in their pathway markers. But I believe here, because the amount of the sample, it wasn't that big, so that's one of the reasons why we didn't detect that many of these lipid mediators from EVs, but we did see them, and also, but yeah, they were in lower amounts than in the concentrates. And we looked at also the biosynthetic enzymes related to the lipid mediators. Here are some examples. So the enzymes related to the pro-resolving lipid mediator synthesis, they were found from the EVs. And so these, so the first one is the standard. The next two are day 1 samples. The next two are day 8 samples. So we could see the, for example, a lipoxygenase and these cytochrome P450 enzymes. And we also saw enzymes related to the prostanoid synthesis. COX-1 was mainly visible in the day 8 samples. So this is like constitutively expressed, but if the cells get like an inflammatory stimulus, then COX-2 expression is induced, but we didn't see the induction of COX-2 in these samples. So most likely the increased production of these arachidonic acid derived pro-inflammatory mediators comes from COX-1 pathway. We also saw that in the EVs there was some of these phospholipases that cut the fatty acids from the phospholipids, and so we So all in all, we saw that the EVs also contain this machinery required for the lipid mediator biosynthesis. Of course, we don't know if they make them themselves, but at least they have the machinery in them. So all in all, the platelet EVs contained a variety of lipid mediators, and in general, the pro-resolving lipid mediators from EVs hadn't been profiled from any types of EVs to our knowledge, and also the biosynthetic enzymes were found from the EVs, and well, we did see an increase in both the pro-inflammatory and the pro-resolving lipid mediators in the bag. Typically, in order to attempt to maintain homeostasis, cells, if the production of the pro-inflammatory mediators increase, they also try to increase the production of the pro-resolving so they could regulate and counteract the very pro-inflammatory signaling. So we think maybe this is one attempt of the cells to regulate this, or they just accumulate during the time. But we should remember that these are quite bioactive and quickly inactivated molecules. Yeah, so what is coming next with the EV lipidomics? Well, in general, the role of lipids as signaling molecules is more and more appreciated in many fields, and there have been some papers that suggest that, well, by doing lipidomics of blood and urine EVs, we could find non-invasive biomarkers in certain diseases. Moreover, we have an increased sensitivity of the mass spec instrumentation, so we are actually better able to profile the samples. And also what's ongoing in the field, like in many fields, trying to harmonize all different setups, and also there have been some lipid annotation issues. So if you just happen to get mass spectrum and you don't really know how to annotate lipids, you might go a bit wrong. So if you ever happen to get lipid mass spectrum, please also ask guidance from lipid people, because we are happy to help. Yes. And so, yes, I'm thanking again everyone involved in all these projects, and I'm also thanking you for your attention.
Thank you, Mina, for the interesting presentation. Now the floor is open for questions.
Okay, I will ask. I remember one work was where he studied, found such amazing observations that if you culture and collect EVs from culture cells, they're quite rich with phosphatidylserine. No, but if you are looking in blood, no. There is no EV purified from blood. They are lacking not such abundant phosphatidylserine, and the explanation was that there was first what immune system scavenge from the blood, and probably by TIM4 interaction with high affinity. Did you observe such in your lipidomics?
We did see there was a trend of increasing one of the phosphatidylserine species that I think you are now referring to, but there wasn't like a significant change in this. But it did seem that the phosphatidylserine was increased, and also in other studies that I work with later, it does seem that like phosphatidylserine in general is quite high from like cultured cell secretome. Yeah.
But you show also that what lipids increased in EV, and phosphatidylserine was in this list.
Yes.
in your presentation.
Yeah. That's well, there that's like a general what is believed, but yeah, I can't say that if it's like, I think it's mainly from cultured cells, these experiments. Yeah.
Yeah. Just interesting that we know that only company in the what they provide us team as pan and antibody, or how they say, that it based on team 4 recognition domain. So it looks like in some samples you still would be selection if you do not have EV3 phosphatidylserine on their surface, you would not catch them. No. Okay.
Sorry. Sorry. So I was looking for the question. So does the change in the EV content in the, you said in the platelets, does it affect the function? Because they are changing, so will it affect the function in the recipient person, for example?
Well, these weren't like tested, but as I mentioned, like because the cells do become activated, so they're not like used in like blood transfusions anymore after 5 days, because they well, of course the cutoff is, it's not strictly 5 days, but there's like well, like safe limits also. But yeah, it might have adverse effects in the patients. But of course, I can't say for sure if it's not just because of these lipids, but it's also like many other factors affecting to that.
Hi, it was quite interesting. So I was wondering to ask you, as far as I know, like except this bilipid layer, there is such a thing as corona. So could you please explain how they are connected? Because I don't really understand how they are like interfering.
So the corona
Of EV. So I mean like this proteomic thing around the EV. If I'm not mistaken, I could be mistaken. Yeah.
So you're asking how
how the lipid layer is connected with that, or it's not.
So the lipid bilayer is the corona of the EV. So it's the one, it's the like thing that keeping the EV together. I'm not sure if I understood your question. Sorry, but yeah.
If the corona is a part of this lipid bilayer, or if it is some entity in addition to the lipid bilayer. I have also nearly the same question. When in your model of the plasma membrane and also the EV membranes, you are showing the phospholipids, bilayers, and everything. But this notion that now they say there is a corona around EVs, that corona has also proteins, or what is that relation? Do you have any view about that?
No, unfortunately not. Yeah.
And Mina, how easy is to do lipidomics?
Well, everything. I mean, I think it's relatively easy. Yeah, people tend to do lots of like proteomic analysis and get all these nice pathways, and that is also possible to do from a lipidomics side also. Yeah, so I think it's like proteomic analysis. Of course, you have to have enough material, but like I said, the instrumentation is becoming better, and so yeah, I think it's very feasible to do also from EV samples.
And definitely you need to have a core facility or somebody that knows how to do it.
Yes, I wouldn't start on my own to do that.
Thank you.
Thank you, Mina, for your interesting presentation, and we have a small token of appreciation for you.
Thank you.
Okay, now we will proceed. We are going to have very short moderation, and it will be moderated by Suranga.
Thank you, Gitme. So it will be a very short one. Maybe we won't be having round of questions like yesterday because everyone is tired, I know. So yeah, so since today we had a couple of interesting presentation lined up, unfortunately we lost one of them, which would have been a very interesting topic to be discussed along the line with the inter-kingdom, communication using EVs. But of course, we had very nice presentation from the nematode, the worms. So actually, today's my task is to moderate and to just induce your minds to have a small discussion on how this the communication happening beyond boundaries. Of course, EVs have been appreciated as part of the intracellular communication, but then when the information come up, like it's not only within the species, but then you have between species, or even now there are enough and more evidence to say there are even inter-kingdom level, for example, from Plantae to Animalia and also vice versa. So it's been picking up the interest of the scientists, and also we do have, as a group, a big interest on this inter-kingdom communications even. Specifically also the host pathogen interactions. So I would like you to contribute, because we are also looking for certain avenues. What should be the way forward? That would be one objectives of this kind of small, brainstormings, I would say, like yesterday. Of course, specificities again may be coming into play, like when we heard from the nematode TV stories, like so. Please, I would like to open up this moderated discussion. Anybody can contribute on the line of actually how the extracellular vesicles communication in different species or even different kingdom-wise will be useful in the future in the science, as well as in finding biomarkers, therapeutics, understanding the different etiology of the diseases, etc., etc. So please, yeah, I think I have.
Perhaps I can contribute a little
great.
bit. Also, my knowledge on that part is probably not as deep as it should. But what I would like to do is to share what I know and what has been published, right?
Yes.
In terms of, can EV communicate between at least two species?
Yeah.
Or are they restricted to one species? So if we look back in the literature, and if we go back, as far as 2007, when they discovered that, microRNA were inside EVs. If you take the Valadis paper, I think it's a Nature Cell Biology in 2007. So they describe exchange of microRNA mediated by extracellular vesicles between mouse cells and human cells, suggesting that it could cross, it was an interspecies, right? So other group, it's not going to be exhaustive, but other group, including the group of Carmen Buchreiser in Pasteur Institute, with who I collaborated, right? We developed an assay to test if bacteria-derived EVs could deliver their content inside human cells, and they did, suggesting that not only they can cross from mammalian to mammalian, but also from bacteria to human cells. So perhaps there is no species barrier, as it's the case for many viruses. And so perhaps it suggests to me that the machinery that is responsible for the exchange is probably conserved through evolution. Okay, so perhaps no, there are some species specificity, but publication, but I'm not aware of them, and I don't know how strong are the proof. So to me, as the best of my knowledge suggests that we have several proof that there is no restriction for species, to the contrary of viruses, many viruses.
Okay, thank you, Greg. Any other contributions? Perhaps I may ask from you, because when I saw your data, especially the microRNA thing, so did you did you find anything transferred back to the mammalian, like in your cell models? I think you mentioned that you did some cell model work. So did you find those things you found in the nematode EVs back in the cells in your models?
Yeah, we're running RNA-seq now, and this is on so plasma samples where we've isolated EVs, and we're trying to detect the parasite-specific microRNAs in the plasma.
Okay.
I'm not sure we will, because obviously it's an intestinal parasite. I think we'll be very lucky to pick it up in the plasma.
Okay.
But it is it's the next step for us. If we find it in the plasma, then, because there are the systemic effects of the worms, it's not just a localized immune response. So we're hoping that the EVs go more systemically as well.
Okay.
But I'm not sure.
Not sure.
Just a quick question to Professor Whitehead. Yeah, were you able to identify any bacterial related DNA or RNA or something in your nematode EV models? Like, and then do you find any special impact over there, or any sort of things, or any microbiome changes or anything like that?
It's funny actually, because a lot of the effects that the worms mediate, we lose if we treat with antibiotics. So they do modulate the microbiota. They increase mucus production, so you get an increase in the Akkermansia, because that eats mucus kind of thing. And we think that's some of the beneficial effects. But I mean, it gets even more sort of complicated because, for example, the trichuris, its specificity to host is actually dependent on the microbiota. So the hatching of the eggs depends on a specific bacteria that it comes into contact with in the colon. So we think that's why that a human whipworm can't infect a mouse, and vice versa. When we've done sort of deep sequencing on EVs isolated, we do find some bacterial nucleic acids. Yeah.
Any more contributions? Seems everybody's tired then. Yeah. Yeah. So you have something. Actually, it's unfortunate that we lost that talk on the plant EV, so it could have been even could have shed some more light on how really the interkingdom level this communication would be happening via EVs.
Like what Greg said, it is very amazing that from one side, we are thinking within one species, sometimes between different individuals, mother and the baby, the work that we do, we find it very, very specific, the way that the EV is used. The other side, when you look between different species in different kingdoms of life, it looks like that extracellular vesicles are a kind of media of life. It's a common language, universal language that is somehow used. And this universal language, for example, in the case of the worms and their host, can completely influence the immune system there and somehow completely make them to some of the host to shut down all the mechanisms, or at least bring toleration. That's also the same that we see with the mother and the baby. Hence, from one side, it looks amazingly specific, at least in the systems that I work on, so within one species. And from the other side, the same system is used to communicate between even plants and humans. The food that we eat, or what we were watching also about the milk, for example, from cows and coming. Hence, one of the issues I would have, and I think quite a lot of people, even when we are speaking here, constantly we say cargo, cargo, cargo. I don't know if all of this is coming from what is inside EVs. Sometimes I'm thinking that we are not thinking very well about what is around EV, or what is that membranes, that lipids, for example, was part of them. And I become more amazed when I see, for example, Tiziana work, when she was showing that they have made liposomes and they put also microRNA in it and it worked. I'm just thinking, is it possible that even liposomes itself, that lipids that you have put there, that are also conveying the message? Nevertheless, what is amazing for me is this to me looks like a language that is used in the nature. And more that we go forward, I think that we would also understand that the universality of this language. And the next stages for us is to understand how this language is spoken, just like the languages, different languages that we have on the planet between humans we are speaking. Probably the same kind of tree of languages is existing within different species, different kingdoms of life. And probably in the years to come, that would be the way that we have to look to learn this language between different texts. That was my 2 cents.
Thank you, Alireza. Any other provocative thoughts that anybody is having just before the coffee break? Yeah, in the absence of anything, so I think we can have a quick wind up the session. So you all are invited for the coffee, and I think rest of the posters will be displayed, right? Thank you very much.
It was a wonderful time, at least for me, the last 2 days, to see all of you here and hear all the science that you are doing. I'm not going to take much of your time already, and we are somehow reaching to the end of the meeting. Just as I said yesterday, all of this is thanks to the fact that you have yourself come. We have had a number of very good speakers, and that is also partially thanks to Greg for putting quite a lot of input into this. Thank you very much. At the same time, yeah, the audience were also very wonderful, especially our younger generation are eager and they are standing up and asking questions and all things. Then hopefully this is actually a sign of the healthiness of the team subject. That I was, I am also the president of the Baltic Society of Extracellular Vesicles, and we had this year in April, we had our meeting in Lithuania, in Vilnius. And I have another colleague, because I come from a reproductive background, that she is actually the secretary of the Baltic Society of Fertility. And maybe I shouldn't say this, she was there because she's Lithuanian. And she said to me, Alireza, it's amazing. You have so many young people here. Our average age of our society is something around 45, 60. And if I look at your guys, they are something around maybe 25, 30. And she was definitely, and she's pointing to a very, very important point. And that shows this is a topic that has a future. Therefore, people are investing in it, coming into it, and are thinking that this is something that they want to continue in future and go ahead. Hence, it's always nice to be involved in such kind of the topics that are evolving and going up. Not that fertility is not good, but people seems that they don't like sex anymore. Hence, that is where we are. Yeah, I'm so, when I make even jokes, I'm so serious making jokes that nobody laugh about it. You are only try to, okay, this was the, then thank you for all of you. And also I thank yesterday also all the guys. I should thank Omid particularly because he probably lost another 5 kilos during the last 2 days by just running around and giving the microphones, everything to you. Should we get a special applause to him? Thank you. Great, perfect, thank you. We had, because of, I would be honest, because of budgetary situations, we had limitations. We had a we have organized going out tonight to do some fishing, and then after that eating the fish together. But we didn't open it to everyone because we didn't have enough money. However, now it seems that we have 30 places and we have only 22 people. Hence, 8 people that come, and this is first come, first serve. Where is Subhashini? Yes, 88 people that, and the priority would be given to the non-Estonian residents, not non-Estonian nationals, but non-Estonian residents, the guests that go to Subhashini. If you somehow reach to her and somehow register yourself, what time we are leaving? At 6:30 in front of the, sorry? Sorry, I didn't understand. Where? Or is it near to the bus station? Is it? Okay, so Ashini will tell you where you have to go. I'm still thinking where is that, but okay. Then at 6:30, we would be going there, and this is something Estonian. I don't think in any other part of the world they make the fish like this. It's a particular type of wood that they would use to make it, and so. As it would be hopefully interesting for you. And if you are interested, jump to Suhashini and do that. And apart from that, I wish all of you a very safe trip back home and hope that you can keep in contact and in touch with each other. Thank you very much.